not only in Pakistan, but in the Islamic world, of how important it is to get a good handle on AI and machine learning, especially vis-a-vis life sciences.
Absolutely, sir. And I think hopefully soon when we'll have mobility and confidence on travel, we would then be organizing lots of these events in other parts of the OIC region also. I mean, we'll be calling you also, inshallah. We'll request you to travel to other places as well, inshallah.
My pleasure. My pleasure.
So, sir, should we start formally now?
Please.
Assalamu alaikum, everyone, all the participants and distinguished guests. So today we are here at comstech-oic ICC International Workshop on Artificial Intelligence and Machine Learning in Life Sciences. So quite a few of participants have already joined the Zoom session. It was supposed to be a physical and a virtual event, but unfortunately due to the rise in cases of COVID, so we have transformed into a virtual event. So we requested all the participants to attend this seminar and workshop virtually. So we'll start off with the opening remarks from Dr. Wasim Haider, who is a professor at ComStat University. So over to you, Dr. Wasim, for the opening remarks.
जी अस्सलाम वालेकुम एवरीवन नहमदु व नुसल्ली अला रसूलिल करीम अम्मा बाद फ़ाऊज़ु बिल्लाहि मिनश शैतानिर रजीम बिस्मिल्लाहिर रहमानिर रहीम रब्बि इश्रह ली सदरी वाहलुल उक़दतम मिन लिसानी यफ़क़हू क़ौली. I would like to welcome all the participants for this very exciting event, for which I would like to thank Professor Dr. Iqbal Chaudhry. The idea behind this was, in fact, they organized an event about AI a few months back, and then I met Professor Saab and I requested, sir, I think we should also need it, the researchers in life sciences, and I think we should also do that. And Professor Saab were very kind, and he said, yes, go ahead and do it. So initially we thought of inviting participants like a conference, but we realized like there are maybe very few people who are applying these techniques in their research work. So then we did some brainstorming, and through our contacts and through Professor Saab's help, we were able to gather around 6 speakers as far as this workshop session is concerned. This workshop session is all lecture based, and inshallah soon when things will be easy, we will also do some hands-on session, most likely in ICCBS Karachi. You're most welcome on behalf of Comstech. Comstech is basically established by the 3rd Islamic Summit of OIC that was held at Makkah, and the OIC Standing Committee for Science in Technology that came into existence, which is now known as Comstech. So headquarters is in Islamabad. The other hosting institute is ICCBS, International Center for Chemical and Biological Sciences. Very wonderful institute. I've been there. I've been attached with one of their sections, this Jamilo Raymond Center for Genome Research. Now this HEJ Hussain Ibrahim Jamal, which is the Alhamdulillah renowned research institute, Professor Ataur Rahman Sahab, he established this, and obviously Professor Iqbal Chaudhry's role was marvelous. Like now this institute has become ICCBS, International Center for Chemical Biosciences, Chemical and Biological Sciences, comprised of HEJ Hussain Ibrahim Jamal Research Institute, the most famous LEJ Latif Ibrahim Jamal, Dr. Panjwani Center for Molecular Medicine and Drug Design, and NIV, National Institute of Virology. Along with that, we do have 4 to 5 more institutes which are under this umbrella. But my favorite is obviously Jamilo Rahman Center for Genome Research, with which we have the state-of-the-art, these next-generation sequencing technologies. I'm working with them as well. In our this inaugural session, we have His Excellency Professor Dr. Iqbal Chaudhry. He is the Coordinator General of Comstech. In the keynote speech, we will have Dr. Saurabh Sinha from Granger College of Engineering and Computer Science, University of Illinois at Urbana-Champaign, one of my teachers. So I contacted with him, and he was very kind. I did my PhD there, and he was associated with the Department of Computer Science at that time. So our guest of honor is Dr. Ghazna Khalid. a professor of public health, women's health, and gynecology. She's an advisor in health emergencies and DMA with the Prime Minister's Secretariat, Government of Pakistan. We are very thankful to her. And Dr. Shahid Mahmood, Chairman and CEO of Interactive Group. He's here with us. Thank you so much, sir, for joining us. Welcome you. In the other sessions, the talks, I will go over with very brief and overview of this AI and ML, and then a little bit about their applications in our research. A couple of our projects are where we applied that, so I will show you guys in that session. Dr. Aziz, he's at Stanford Cancer Institute, Stanford University, USA. So he'll be talking about the applications of ML in regulatory genomics and epigenomics. Then we'll have the other session where we have the talk from Dr. Ahmed Raza Shahid, one of my colleagues at ComSAS University, Islamabad. He's developed the application for this brain tumor segmentation and survival prediction. So basically, it's a software where you give the images and it predicts cancers in them. Then we will have Professor Dr. Mohammed Shafiq from New York University, Abu Dhabi. He will talk about the efficient deep learning for healthcare and bioinformatics, trends, challenges, and opportunities, followed by the last talk of today's session by Dr. Adil Razi from Turner Institute for Brain and Mental Health, Melbourne, Australia. He'll be giving an overview about the computational neuroscience, brain models, networks, and inference online. And then our session will be closed at around 3 p.m. So thank you so much, everyone, for being with us. Hope this exciting session will be good for all of us, and we'll have the networking and sort of brainstorming activities for all of them. Sorry, I realized my video was not on. So yeah, so looking forward, and hopefully it will be a very interactive session. So we'll have the lot of exciting people around, and we might think of going from here, because this is our first effort here, and obviously we'll learn from it, from our experience and from the guidelines from our elders, and inshallah we'll go further from this. Thank you so much, and welcome everybody, and over to our host.
Thank you, Dr. Wasim, for your opening remarks. Now I would request Coordinator General Comstech, His Excellency Professor Dr. Iqbal Chaudhry, for his welcome address.
Bismillahirrahmanirrahim. Assalamu alaikum. Good morning and good afternoon, ladies and gentlemen. We are in different time zones, and I'm sure that as day pass by, we will have more participants from the western side of Pakistan, because a lot of our member states are located at the west and north, and that would require some time before they join us. But I'm sure it's going to be a major event, and this inauguration itself actually represent wonderful things. For instance, we have Professor Dr. Shahid Mahmood, a very dear brother. He is a man par excellence, person who represent the best of human attributes and excellent talent. He has been working tirelessly in Pakistan for promotion of science and technology, and has a lot of contribution in the region also in promotion of science, technology, culture from in different levels. So he represent industry, he represent policy making at different levels, and he represent the international advocacy for science and technology. We have with us Dr. Ghazna Khalid, their sister. She has been instrumental in making lots of fundamental changes in the policies of government. So she has been instrumental in policy reform in the healthcare sector. She's a practitioner of healthcare, has lots of insight about what is required to be done in healthcare, especially in the context of a developing world. Wasim, Dr. Wasim Haider has already introduced you to this event. He is a young, promising researcher from an academic institution. So certainly you can see the nexus of industry government and academic institution. Along with this, we also have Comstech as the host institution. And as Wasim mentioned, Comstech was established in 1981. It is the collective, it represents the collective will of 57 member states, the second largest bloc of countries after United Nations, and an extremely important one because these are the countries with the highest increase in population, with great prospects of development, and a lot of Africa, major part of Asia, and some part of Europe and South America. The countries lie in these regions are part of the COMSTEC. So 57 member states, 57 ministers are the member of COMSTEC, which is also responsible of the implementation of science and technology innovation agenda STI 2026, which is equivalent of SDGs, Sustainable Development Goal. And one of the key factors in which STI addresses planning and the need of implementation of technologies is emerging and disruptive technologies for human welfare. And certainly AI is extremely important. We have seen what has happened during COVID-19. We have seen AI and machine learning used in patient care, in hospitals. It was used in tracking and tracing of COVID-19 positive patients. And most importantly, it was used in analysis of big data. This tremendous data which is generated through genomic, proteomic, and other biological research was made available to people by using machine learning. And that is the reason why we were able to do lots of wonderful things. So it was a remarkable year for science. But I would say that science was able to do that. Laboratories were able to perform only because there was enabling technologies, and AI was certainly one of the most important technologies made available to scientists. And we have seen what has happened, that within 6 weeks of the emergence of disease in Wuhan, the genome was made available to the people. It was originally published. Now we see literally hundreds of thousands of genomes of virus available, constant monitoring of changes in virus, so tremendous data generated, which was made possible only because of machine learning. And we can analyze this data much more efficiently by using the AI tools. And because of that, we were able to develop diagnostic tools. Because of that, we were able to decipher the complete biology of the virus, and that help in devising right policies, from social distancing to mask, from understanding the surface viability of virus. There's a whole range of different development took place, and science was able to deliver only because of the presence of these wonderful enabling technology. And I think the most important one eventually at the end was the vaccine development. So it was all possible because AI and machine learning was effectively used in deciphering the pattern trains and data which was available to scientists. And this is why it is important for Comstech to work towards that direction. And we have been extremely fortunate to get the support of Dr. Shahid Mahmood and Dr. Razna Khalid in this endeavor. We have organized several events in the field of AI and its application in health. We are in the process of establishing a research center, particularly focused on the application of artificial intelligence in health. So a number of things are actually happening. And as the mobility facilitated, as we have more confidence in traveling, we would take these events to the other countries in the OIC region. So we intend to organize events in West Africa, Central Asia, and South Asia. In all three different regions, we intend to have this event organized, which include exhibitions, workshops, and other capacity building initiatives, including research fellowships. With this, I would not really like to take much of your time. I'm sure that you're excited to listen to our expert from USA, and I'm sure that they would be able to give you much more useful information of what is required to be done in terms of developing capacity and at the interface of biology and computer science. And that is where the most exciting thing happening nowadays. Uh, I am extremely grateful to Chief Guest and Guest of Honor. I'm grateful to Dr. Wasim Heda, Haris, and Dr. Ayaz for putting together an excellent program. And I'm absolutely delighted to welcome you all in this event. And hopefully soon we'll be able to meet in person. And we'll have many of such events in Islamabad and other places of OIC region. Thank you very much. Thank you.
Thank you, sir, for talking about the programs, different programs of Comstech. Now we'll move on to the keynote speech. Dr. Saurabh was supposed to have, we would have him online with us, but due to the time zone difference, he shared the recorded video, and I'll just play his video from here.
We can't hear you, Haris.
Haris, speaker is not audible, so this video need to be.
I'm just checking.
Yeah.
Hello, I'm Saurabh Sinha from the University of Illinois at Urbana-Champaign in the US, and it is my great pleasure to be talking to you today. Today, I'll present to you an overview of the NIH-funded Center for Excellence for Big Data to Knowledge, or BD2K, that I directed from 2014 to 2019, and which tackled the challenge of building powerful tools for genomics. I'll start by pointing you to this perspective piece that we wrote in 2015, where we recognized the amazing growth of data in genomics and predicted that soon genomics may become the biggest source of data for analysts. The funding agencies in the US are, of course, well aware of that and, in fact, pushing for this very vision and preparing the research community for it. For instance, there is considerable buy-in for the idea of FAIR data and software, where FAIR refers to findable, accessible, interoperable, and reusable. And among other ways to achieve the FAIR vision, there is a push for cloud-based software portals that can interoperate with each other. It is precisely this vision that drove the center that I'm going to talk to you about today. This BD2K Center was hosted at the Institute for Genomic Biology at UIUC in partnership with the Mayo Clinic at Rochester, Minnesota. I co-directed it with my colleague, Professor Jiawei Han, and I led the research team of the center. Our goal was to build a knowledge engine for genomics, from which the center got its name, Knowing. The main deliverable of the Knowing Center was the eponymous Knowing platform, a cloud-based portal for genomics data mining. The idea was that genomics data sets are often in the form of spreadsheets, where rows represent genes and columns represent different conditions or samples under which the high throughput measurements have been made. The platform for analysis of such data is hosted on the Amazon cloud and allows the user to upload their own spreadsheet of genomics data, just as you would upload your photos to Google Photos, let's say. And then the user can run powerful analytic tools through the platform on the cloud. The tools offered by the Knowing system address a variety of popular bioinformatic tasks such as sample clustering, gene set characterization, gene prioritization, phenotype prediction, signature analysis, and so on. And these tasks can be useful in answering common biological questions, such as what subtypes does this disease have? Which biological processes are these genes involved in? Which genes are associated with this phenotype? How long do we expect a patient to live? And so on. And the tools provided relied upon a variety of methods from machine learning and data mining. One of the highlights of knowing analysis includes new tools for knowledge-guided analysis, whereby existing genomics knowledge bases or databases represented as a massive heterogeneous network are brought to bear upon the analytical operations provided to users. For this, we encode into a graph format the data from a number of genomics databases. These include pairwise relationships among genes, such as protein-protein interactions, genetic interactions, and homology, and so on, as well as annotations of genes, such as pathways, gene ontology, etc. The nodes of this graph will thus be the genes and properties, and the edges will represent the known gene-gene relationships and gene property annotations. And just to remind you, there's a lot of public data to encode, so the graph is quite large, with hundreds of thousands of nodes and 500 million edges. Now, here's the underlying idea of knowledge-guided analysis or knowledge network-guided analysis. You have this spreadsheet on the right with genes as rows and biological samples as columns, a typical input for machine learning algorithms. But you also have this separate graph from prior knowledge, where the nodes are the features of your spreadsheet. And the edges, therefore, represent relationships among these features. How can you exploit this graphical knowledge in improving the accuracy of your machine learning tools? Note that if there is one thing we've learned in 3 decades or more of bioinformatics, it is that prior knowledge and context really matter for machine learning performance. So this was the central algorithmic question driving our work, the use of knowledge network in the machine learning analysis of user spreadsheets. There's also an obvious cyber infrastructural challenge posed by knowledge-guided analysis. There's a massive knowledge network involved in the analysis, and we don't want users to have to download this to their computer and keep it updated all the time in order to run our machine learning tools on their data. Plus, some of the analysis tools may be prohibitively compute intensive, making local execution of the software not such an attractive prospect. The solution we pursued, therefore, was to have the knowledge network and the tools co-localize on the cloud, and the user simply moves their smaller data sets, the spreadsheets, to the cloud prior to analysis. We put together a strong software engineering team to make this happen, thanks to our partners at the National Center for Supercomputing Applications, or NCSA, and they made sure that the tools made by our graduate students and postdocs were suitably packaged in Docker containers and plugged into a Kubernetes orchestrated system for automated deployment and scaling. So this is what the data flow looks like for analysis at the Knowing platform. The researchers get their omics data from the biotech or sequencing center next to them, hopefully already formatted as a spreadsheet, and then they send this spreadsheet to the cloud where the knowledge network and the Knowing tools or pipelines already reside. The analysis is done at a click of a few buttons, and the results are sent back to the user. But there are other ways in which the Knowing tools can be run in the cloud. For instance, there is a cloud-based ecosystem of data and tools for cancer genomics, the CGC, or Cancer Genomics Cloud. That's this pink shaded cloud. The user may be doing their routine analysis on CGC datasets and say they want to run a Knowing tool, this one, then and there. For this, we have published our tools on the CGC, meaning that the tools are available for use on the CGC cloud, and they can seamlessly access the knowledge network, which resides in the Knowing cloud, thereby achieving knowledge-guided analysis without the user worrying about the logistics of moving the network or their data around. In the remaining time, I will briefly outline some of the bioinformatics approaches, the machine learning techniques that were developed by the Knowing Center. These pertained to the challenges of incorporating the knowledge network into the analysis, as well as to extract novel insights from heterogeneous multi-omics data. In one project, we were interested in identifying genes associated with drug resistance based on gene expression and cytotoxicity data on large panels of cell lines. Note that one can measure the sensitivity of a cell line to a drug, i.e. the cytotoxicity, by experimentally obtaining a dosage response curve such as this, and then summarizing it into a single number. Our collaborators at Mayo Clinic had such cytotoxicity data for many different drugs on cell lines derived from 300 different individuals of 3 different ethnicities. Moreover, gene expression data were also available on those cell lines. The simplest approach to identifying genes related to drug response variation among those individuals would be to use correlative methods between expression and cytotoxicity. But we showed the power of knowledge-guided analysis here. We transformed the gene expression matrix to incorporate the knowledge network using random walks with restarts or network diffusion, and then correlated the resulting transformed expression profiles of genes with the cytotoxicity profiles of cell lines to rank the genes. We showed that this clearly improves the ranking of genes compared to regularization-based approaches, which is what might come to your mind first, such as ElasticNet, without the use of knowledge networks. The top predictions of cytotoxicity-related genes were then experimentally validated by our collaborators. Another challenge where such a network diffusion approach made a big difference was that of sample clustering, where the goal is to cluster the mutation profiles of patients into groups in the hope of discovering subtypes of a disease. We adopted an approach published earlier by the Idekar lab at UCSD for this purpose, where network diffusion is used to smooth the mutation profiles of patients before clustering them in using non-negative matrix factorization. The result is a powerful sample clustering tool where you upload a mutation matrix of genes by patients, and it will group the patients in a way that distinguishes different subtypes. So each of these blocks here of a different color is a different group of patients that was discovered in the cancer mutation profile data. In a different project, we tackled the challenge of defining gene signatures, which is a concept quite popular in pharmacogenomics and cancer. In particular, we asked if the transcriptomic signatures or transcriptomic changes due to a gene's knockdown in a cell line truly form a signature. We analyzed the Links L1000 compendium of hundreds of thousands of such transcriptomic profiles and set up a classification task of predicting which gene's knockdown is represented by a given before and after view of the transcriptome. We used a convolutional neural network for this task and tested it on a 400-way classification task, where the expected accuracy is a mere 0.25%, and we achieved a 70% accuracy for this task in predicting which of 400 possible gene knockdowns the given transcriptomic change represents. In another project on cancer pharmacogenomics data, we used a probabilistic graphical model to integrate expression, genotype, i.e. EQTLs, epigenotype, i.e. EQDMs, and drug response data on a panel of cell lines, along with transcription factor ChIP-seq data from the ENCODE project, to identify major regulators or drivers of drug response variation across individuals. Again, our collaborators validated several of our predicted regulators via knockdowns in cell lines. In another work, we used a probabilistic graphical model to identify transcription factor gene regulatory relationships that especially matter to a given phenotypic variation, in this case, the tissue of origin of tumors. Developed fast methods to identify pairs of genomic features for separating two classes of objects, such as up and down regulated genes, achieving over 400x speedups. Developed novel ways to identify pathways associated with drug response using network diffusion again to represent genes and pathways in the same low-dimensional space, and made some progress on using models trained on cell line data to make drug response predictions on patient cohorts from TCGA. The culmination of those 5 years of work on building tools in cyber infrastructure is the Knowing platform, which allows users to run our tools in the cloud. In ending, I'd like to say that the genomics community has really got its act together in moving towards a fair world with easy-to-use tools and data, and our work was a small contribution to this community effort. Machine learning and AI will clearly play a key role in this analysis, given the high dimensionality of the data, the large volumes of training data, and the interrelationships among different components of the data. Large specialized databases are a trend, and these databases tend to be on the cloud these days. And the future tool development should be keenly aware of the need for interoperating with these cloud-based databases. Prior knowledge is key to achieving greater generalizability from machine learning techniques for biological data, and rigorous algorithmic approaches are necessary to incorporate that prior knowledge. And the grand challenge will therefore be to facilitate multi-level mechanism-driven analysis of multi-omics datasets informed by knowledge bases of molecular properties and interactions, and all of this on the cloud. I'd like to thank the NIH and an outstanding team of researchers, scientists, and software engineers at the University of Illinois and the Mayo Clinic for making this work possible. And I'd also like to thank Dr. Wasim Haider for inviting me to this fantastic symposium today. I hope you have a great day. Thanks.
We hope things get better and we can invite these international experts to our physical events in the future. Now I would request Dr. Ghazna Khalid, our guest of honor, to address the participants. Dr. Ghazna.
Assalamu alaikum, bismillahir rahmanir rahim. Thank you everybody for attending, and thank you Dr. Iqbal and your team at Comstech for making this really important event possible. Hardis, could you share my screen for me, please? Could you put up my presentation? I'd like to make this very interactive and personally driven. So I had put together a few slides to talk about the presentation and to talk about the implications of artificial intelligence and machine learning. So this slide is 14 years ago. This is my daughter, 4 years old, doing a stimulated robotic surgery. This is what the future brings for machine learning and automation, and how we can link it to patient care and the life sciences. This is what it really brings home. And so far we've really advanced. This is, as I say, 14 years ago. And we need to look at the personal aspects of how this affects patients. Next slide, please. So as my colleague and Dr. Iqbal have just alluded to, and Dr. Aziz in his previous lecture, artificial intelligence can help doctors make better clinical decisions, and I would say help doctors make judgments to better areas of healthcare. So if you see this slide, we've been shooting in the dark, and Dr. Aziz in his lecture just previously very eloquently put together the individualized area of genomics and cancer genomics and patient care. And this is exactly what happens. When you have big data sets, you can help to individualize patient care by looking at genetic markers. And big data sets and artificial intelligence and machine learning can help prime those. Could I have my next slide, please? So we've looked at cancer genomics. Dr. Aziz, in our previous lecture, has just given an overview of that, how that can help. But what does this mean for patients? What does this mean for to help people to get better care? Yes, we know about genome-wide association studies. Yes, we know about the genetic mutations that make people either susceptible to different biological agents, different pharmacological agents. But what does that actually mean for patient care? Could I have my next slide, please? So this is Moore's law, and this is how what has helped develop in the recent years of what does it mean for patient care. We're individualizing patient care. We're using our big data sets to individualize patient care, and eventually we will have our genome on a chip, which will help prime using that big data to know the susceptibility, not only to cancer, but also to other non-communicable diseases, like diabetes, like asthma, like blood pressure. And we will be able to know and predict from our genetic structure the susceptibility of those genes. So this is why artificial intelligence and machine learning is so important to actually prime care. So I was delighted when Dr. Iqbal asked me to just give a brief overview of why I feel, from a personal perspective, what is the cost in terms of human lives saved and in terms of the contribution to physical sciences. How will it actually impact? Could I have my next slide, please? So, as I said, genome on a chip. And this is going to be really possible in about, less than $1,000. And I'm sure my learned colleagues would, educate me further from the artificial intelligence perspective, but certainly from the medical perspective, this is so much needed and will determine and tailor-make patient care and then be the next revolution in the life sciences. Next slide, please. So Dr. Aziz has just talked about in different pathways the whole genome sequencing of tumor, and it just gives an example of why we feel that data sets and machine learning and artificial intelligence is extremely important. It is the reference laboratory. It's basically having a library of data like the 100 Genome Project, and it gives you the data set of how different genomes are predictive of patient care and patient susceptibility to different medicines, different drugs, and different modalities of treatment. Next slide, please. So we've talked about tumors a lot because it's something that is close to my heart and it's something that I've done a lot of research on, but we're going to create the black box. We're going to create translational intelligence and identify the risk of relapse and the need for treatment and identify gene signatures predicted of response to cytotoxic agents, as Dr. Aziz has just alluded to in his previous lecture, and specific targets of biological agents. And we won't be shooting in the dark anymore. Cancers are not a homogeneous in modality. They're different entities branded together. And so once we know and we can use our big data sets, creating genome-wide association studies, we'll be able to better treat our patients and have personalized care. Next slide, please. So the future for doctors is artificial intelligence and machine learning. And next slide. And eventually people will have their genome on a chip, and that's the future for our patients. Next slide. So this is coming closer to home in terms of the COVID-19 and looking at where artificial intelligence and machine learning has helped assist us in this devastating pandemic. And as we all know, we know the horrible figures. 1.6 million people have died approximately, and 70 million have been affected. And we have used artificial intelligence and machine learning for traditional to assist and to help with epidemiological tools. And Dr. Iqbal and I sat on the COVID-19 task force. We looked at different areas of locating and GIS data sets to assist in helping track and trace patients, and that gives a good model, and that's been used in different parts of the world. The artificial model systems improve pattern recognition of disease and different populations, and gave mathematical models of where we felt that the disease would spread. So it helps public health officials select better response and preparedness against the COVID-19 pandemic. Next slide, please. And that then brings up to disaster management as a whole. And our country, Pakistan, is, as you know, prone to disasters, earthquakes, floods, now the pandemic, and AI-powered digitalization of disaster management will help reduce the risk to life, minimize the losses, and mass casualty incidents will be assisted by prompt and appropriate responses to these disasters. So we can use sensors for real-time situation awareness. We can use the integration of wild information on weather prediction, and we can automate the processes of handling a disaster, both pre, intra, and post-disaster. This will all lead to the reduction and the loss of precious lives. So we need to have an early warning system based on sensor data. Next slide. And this I felt was particularly interesting, and I got very excited by use of artificial or augmented reality platforms and mass casualty incidents, MCI, and where you can use smart glasses, Google glasses, provide real-time data, real-time video and image transmission to remote viewers, and provide incident data to the stakeholders, the doctors and paramedics at hospital sites, and the allocation of hospital resources, triaging, transport to better care for patients in these aspects. And I think this is really pertinent to Pakistan. This will be pertinent to our partners in the OIC countries. If we can create such augmented reality platforms, it would really help to save the precious lives. Next slide, please. So I'll finish off by giving this brief overview by saying that artificial intelligence and machine learning is the future, is already here, and we need to intertwine it into our patient care and to tailor our patient care to provide better quality and informed decisions and understand disease processes. And in any case, the glass is always half full and the future is bright. Thank you very much.
Thank you, Dr. Ghazna, for sharing your wonderful thoughts. Now I would request our chief guest, Dr. Shahid Mahmood, for his address.
Could you allow me to share my screen because I need to upload a presentation?
Yes, sir.
Is it visible?
Yes, sir, it was visible.
Is my presentation visible?
Not yet, sir. We did saw a screen of Word document.
Just a second. Sorry about this. Now?
Not yet, sir. Yes, sir, now it's visible.
Oh, this is another. Sorry about this. Why is it not? Okay. So let me not waste time on trying to open my presentation slides. I can sort of try and cover all of this as an extempore. Very grateful, Dr. Iqbal, for allowing me to speak at this very critical juncture and hosting something which I believe is very, very important for not only Pakistan, but the Islamic Ummah. And as you are currently looking after comstech also, it is very critical that we look at a few aspects. Very well thought through list of speakers. I've seen the areas that they're covering. Dr. Sinha, I think, showed us where technology is taking us, where the real world is working, and what we are doing and what we can do. So let me take this to another level. I think it is critical for us to be speaking at a level that sort of sets the roadmap for not only this workshop and this seminar and the discussions that we will have today, but for the future course of what comstech should be doing and what we all should be looking at as our future challenges. Step one, we are very clear that by 2100, the world's population is going to be anything between 8.8 billion to 10.4 billion people. That is adding another billion people every 10 to 12 years, and then settling at a certain point. So the two predictions are 8.8 to 10.4 billion people. That's one side of the equation. The other side of the equation is that all of this population base is going to shift in two aspects. One, 40% of this population will be in Africa, and 42% of this population will be in Asia. When you look at these two numbers, and then you look at how these changes will take place, let me just point out the top 10 countries in 2100. And I do this as a quiz question to my kids and my grandkids, that do you really understand where the world is going? Because as scientists, as people who have some knowledge, it becomes fundamentally important for us to steer the right direction for our knowledge gathering and for our future analysis and work. So the first country in the world, which is China right now, will actually not be the first country in the world, and it will be India at 1.9 billion in 2100. And the second country, which a lot of people find as an amusing surprise, is Nigeria. And after Nigeria, the shift in population base would be that there would be about 700 million people in Nigeria and 700 million people in China. And the top countries in the top 10 countries in the world would be countries like Ethiopia, Congo, and others, and including Pakistan. So when you look at all the studies that we're doing today, we then have to understand that AI and machine learning are the tools that are now fast moving, because the processors, the storage capacity, the connectivity is now doubling every 12 to 18 months. So when we have technology moving at that pace, And we have a population shift where all these analysis, especially for future disease monitoring and future control mechanisms and where we are applying this knowledge is also shifting. There is a third factor that is shifting. And that third factor that is shifting is that we are moving from our rural communities to urbanized communities. And by 2100, 85% of the world will be urbanized. Now, when you look at urbanized world, shifted world as a population base, completely new set of people where research is currently not being done. I mean, I would love to see papers coming out of Ethiopia, which is one of those countries which is going to be there. I would like to see papers coming out of Republic of Congo, Tanzania. These are areas where we need to focus because Egypt, because this is where the population belt will shift. And this is where more and more research, Pakistan in itself, we will be the 5th largest nation in the world. While all of this is happening, the data that is being generated, because connectivity is now coming into place. If we take the 3G, 4G phenomena, we are already in the 5G era. So 3G to 5G is like 100 times faster. And by 2030, we will be in the era of 6G, where we will have 1,000 times connectivity speed. So connectivity speed, shifting population, and the geographical footprints are changing. When the geographical footprint changes, it also changes the impact on the various communal diseases and all the stuff that we're trying to study with machine learning and AI as a tool, because manually it'll become impossible. So the world population is moving to something like 8.8 to 10.4 billion. All of these changes are taking place. Connectivity is taking place. And based on the connectivity, when you look at the number of languages that are spoken, because they are the ones that generate the data, because they are the ones who have the local knowledge, you will be amazed that a country like Papua New Guinea has over 800 languages that are spoken. A country like Pakistan has about 79 languages that are spoken. So if we take the top 10 countries, and I'll share my presentation, I'll send it across. I should have sent it earlier, and we would not have had this technical glitch. This will become the challenge. Now, once we set this challenge, I think what fundamentally we need to look at across the board in all, not only in Pakistan, but in the Islamic world, is how will we manage what Dr. Sinha was pointing out as cloud and all the tools that will be made available for all the new research. What will be the cyber sovereignty and the authenticity and of management rules that will apply of the data that will be generated from all of these studies? Because this then becomes the new challenge. Because based on this data, we can now see the race on which is coming to COVID, which is the vaccine, what is the time frame of the vaccine. A country like Pakistan, for instance, I've been discussing this with Dr. Ghazna, that a country like Pakistan, where if we take a rough number that we have 10 people in a home, we normally have 6.7, we need at least 25 million people who are the breadwinners of this country to be vaccinated. And if the vaccination is a 2-dose and has to be done every year, and its impact comes in about 3 months, this cycle has to run. Now, what is the price of that vaccine? How is it administered? Where we've done only testing of under 1 million people. How do we do all of this? Because if we do not, then we go into the vicious cycle of what is called health shock. So this is where machine learning, big data comes into the next focus. That once this data is generated, all of the knowledge that we have, one, what are the processes? We understand, and I think Dr. Wasim Heather has put a good set of speakers today. And with the input of all the speakers that we will get, the second question will come, how do we streamline the data in a manner that can translate into something that it can ethically be managed as the future course of action? And this, I think, is the challenge not only for Pakistan, but for all the states under comstech, the Islamic states in the OIC, that how will we manage all of this when we know that what will happen in the next 79 years and what one COVID in itself has opened up. We have used track and trace very intelligently. Science diplomacy will then become the key word. How do we connect all of our key thinkers in the respective states to understand that whatever we are gathering is ethically preserved in such a manner that it is given to the right set of people? And when I say the right set of people, we need to understand that we are now in an era, and thanks to what we're doing even on Zoom today, we have, from my count, 72 participants listening to this across the world. How do we all collaborate with our respective knowledge bases, minus the ego and minus the supremacy of I know more or I know better? In what I know, how can I contribute towards the bigger picture? And the bigger picture would be that how life sciences can be made more accessible and more advanced so that we can do better for humanity. And when I say better for humanity, bear in mind that we are depleting the Earth's natural resources at a pace of adding 1 billion people every 10 to 12 years, and therefore have to survive within the same Earth's resources. And if we have to share those, and we have to preserve those, and we have to manage those against the climatic changes, because when you move into urbanized situations, as we are moving already, we've crossed 65, we'll be going to about 85, the disease patterns, the pandemic or the epidemic patterns, and all of that will be now being registered with the food intakes that we will have. How will all of those things change? And as we know today that these things can be, how do we develop the right silos of knowledge, which is not only the technical knowledge of how better we understand AI and all its tools, machine learning and all its wherewithal, that helps us in deciphering the data that we have gathered. And it is the processes that I think, as Pakistan, the first thing we need to do, and under our scientific community, is how and where do we store all of this data? How and where do we tell people how to generate this data so it is in a digital format, so then the analysis can take place? And then how do we learn from the international world of all the tools that are available today on how to apply them and then do better? Because let's not forget, we will be the 5th largest nation at about 245 million people in the world, and therefore we will have a significant role to play. And if we take it in other numbers, we will be the conduit to joining Asia and Africa. And that's where it makes it very critical for people in our part of the world, especially in Pakistan, and in the community that we're sitting in today, thanks to you, Dr. Iqbal, and Dr. Wasim, that you've gathered people from different backgrounds, different understandings, to be able to do the analysis and share the value of the knowledge that we already have and how we can take it further. I would only add that during your conclusion sessions, you should also add what would be the next steps in the things that I've just highlighted. With that, I would thank Dr. Iqbal and Dr. Wasim Haider, and it was indeed a pleasure to listen to Dr. Ghazna and Dr. Sinha in his opening keynote address, very, very insightful. And thank all the speakers that will follow for contributing towards this an amazing session. And I wish you all the very best. Thank you.
Thank you, sir. Thank you.
Thank you, sir. On behalf of Comstech and the organizing committee, I thank Dr. Shahid, Dr. Ghazna, Dr. Saurabh, and Professor Dr. Iqbal for taking out time for the inaugural session of this event. Now we'll move on to the technical sessions of this event, and we'll start off with Dr. Wasim Haider. He'll be introducing and giving the overview on artificial intelligence and machine learning for life sciences. Dr. Wasim.
Yes, please. Let me share my screen. Assalamu alaikum, everyone. This is Dr. Wasim Haider. I'm assistant professor at Computational Biology and Bioinformatics Group. We abbreviated it as CBBG in the Department of Biosciences, ComStats University, Islamabad. And I'm also having a team, we call it Next Gen Solutions. Next generation sequencing data analysis is done here. And the name is actually having two meanings. The other thing is it tries to help the solving the problems of our next generations. Like we provide opportunities for internship. We give financial supports to needy and deserving students, as well as we do some other community services work with our profit. So this organization is all dedicated for community services. Also helping Jamilo Raymond Center for Genome Research whenever and wherever they need my help or consultation regarding this next generation sequencing data analysis. My talk is divided into these following components. Actually, my thinking was like, since we have the people coming from various backgrounds of biosciences, life sciences, as a whole, like agriculture and medical sciences, and we were expecting some people coming from computer science background as well, and some from engineering as well. So it's kind of a very generic talk, like a lecture, a classroom lecture. So that might help you guys understand the things coming in the lectures of our next speakers. So I'll try to make a way out and make it simple and easy understanding of the basic concepts coming both from genomics and as well as the other aspects, transcriptomics, and a little bit from proteomics as well. So various domains of life sciences where we can apply these AI and ML techniques, and then a brief overview of these AI and ML techniques as well, and followed by uh, some work which we actually did in our research group, though not it's in infancy, but obviously it will help you make an idea of how and where we can apply this thing. So we applied in an RNS sequencing data. Comstech, thanks to them, basically this, let me put this thing on a side. How can I do that? This, yeah. So Comstech is established by the third Islamic Summit of OIC that was held in Mecca back in, okay, let me go back. So that was held in Mecca, Saudi Arabia in 1981. The core mandate of Comstech is to strengthen the cooperation among OIC member states and this cooperation in science and technology, and enhance their capabilities through trainings and in the emerging areas. So like we are getting benefit of this core objective, and thanks to the organization, thanks to the all participants and the speakers who spare their time for us, and thanks to Comstech for providing us this platform. The headquarters is in Islamabad. The other partner organization that is uh, collaborating in this event, and obviously we will be doing some hands-on training later on at ICCBS, that the International Center for Chemical and Biological Sciences. Everyone is familiar with Hussain, Ibrahim, Jamal, HEJ Institute, obviously world renowned institute. Professor Ataur Rahman Sahab worked there, and Dr. Iqbal is working there and heading that institute as well. So this is Hussain Ibrahim Jamal Research Institute, and later on I was told when I was doing a training over there that that was a generous family, and I think these two are brothers, Latif Ibrahim Jamal Sahab. So they funded for the building and some of the resources. LEJ is I think the IT resource are the libraries there and the digital libraries there and the lecture rooms, and these facilities are there. They also have a National Institute of Virology and Dr. Panjwani Center for Molecular Medicine and Drug Design, which is also world-renowned institute in molecular medicine and drug design. And there are, I was seeing on the internet, there are 8 other institutes as well. So total, there are these 12 institutes that come under the umbrella of ICCBS. One of my favorites institute there, Jamilo Raymond Center for Genome Research, that's located at ICCBS Karachi. And this was established by Professor Ataur Rahman Saab. In fact, he gave his award money for the building, and later on they got it, and they wanted to name on his name, but he said maybe you should name on his father's name. So it's Jamilo Rahman Center for Genome Research. That has state of the art these next generation sequencers. They had this ABI solid sequencer. They have this NextSeq and MySeq and lot many more things coming in, along with a wonderful computing facility, the high performance computers, HPCs. They are establishing them. So one of the leading institute of the country in genome research, I would say. I belong to ComStats University, Islamabad. So ComStats is another sister organization to Comstech. I just came to know by after Professor Iqbal's talk that Comstech has 57 member states. So in this way, I think Comstech is the bigger organization. ComStats has around 40 member states. So ComStat stands for Commission on Science and Technology for Sustainable Development in the South. In 1994, various representatives from the southern hemisphere countries, mostly these are the developing countries there. So they gather around and they establish this, and the headquarters is there in Pakistan. These are the various member states. I highlighted few of them. The headquarters is again in Islamabad. So this institute decided to come up with the Center of Excellence in Information Technology, IT. So they established CIIT, ComStat Institute of Information Technology, back in year 1998, the Islamabad campus, followed by Abbottabad and Wa and Lahore. And total now we have 8 campuses with 1 virtual campus and 7 physical campuses. This then institute transformed into ComStats University Islamabad a couple of years back. This is my Department of Biosciences. Lovely place. We have 100 plus faculty members, with which we have 86 are PhDs, and we work in 9 different themes, and these are our various themes, and I belong to this computational biology and bioinformatics. This is our CUI's bioinformatics faculty. Dr. Fatmi, our team lead, he's also ex-student of Professor Professor Iqbal Chowdhury. He did his MPhil with Dr. Iqbal at HEJ, and did his PhD from Austria, and then postdoc from USA, and Dr. Rauf, and PhD from France. And we have 2 Korean, one from China's top university, Inha. And then we have 4 of us from University of Illinois Urbana-Champaign, and one local PhD is also here, but she did her research work at Max Planck. This is a one slide about me. I'm a PhD from University of Illinois Urbana-Champaign, and I also had a master's degree in computer science back in year 2008 from Barani Institute of IT. Before that, I did MSc and MPhil in biology from Quaid-e-Azam University. My research interests are high throughput sequencing data analysis, statistical and computational genomics, and applied statistics, and software development. And sometimes we also do this web and database development. Okay, so that was about me. Now this is about DNA. So everyone knows, or most of you know, what the DNA is. It's the heredity material that controls our life, that controls our heredity, pass on to the next generations. This is how it looks like. So everyone knows that our living machinery is made up of the cells. So within the cells, we have this central part called as nucleus, very important. So this nucleus contains the chromosomes. So chromosomes is actually a bundle or a packet of the DNA. So DNA is actually packed in these chromosomes, right? So if you go from here, we see this is a actually double helical structure. These are 2 threads which are wrapped around each other, right? So there is the red one and there is the green one. This is just for demonstration purposes. Within that, we have these ATCGs, some alphabets within this DNA. So actually, our interest is to play with these alphabets, A, T, G, and C's. So what are these? Basically, these are the biomolecular structures we call them as nucleotides. I will come towards them in a moment. So DNA is attached with some proteins called as histones, and then it gets more complicated and more convoluted, and finally we reach here. We see that it is coming out from this structure. We call it as chromosome, the packets, right? How many packets are there of the DNA in a human cell? We have 22, what we call them as the autosomes, and some people say this is 23rd and this is 24th, but 23rd pair we can say is belonging to the sex chromosome. So we are all similar in all these chromosomes, the autosomes. So these are all the autosomes, and then these are the sex chromosomes where male and female are different, right? So boys, this is the thing that makes you different. This DNA is actually packed in these chromosomes, so that DNA has a distinguishing feature. Within the DNA, everything is the same, but the difference comes from those nitrogenous bases. So nitrogen-rich compounds, we call them as nitrogenous bases. They are there. So we have those double ring nitrogenous bases called as purines. One of them is adenine, one of them is guanine, the double ringed. And then we have the single ring molecules called as pyrimidines. So these are C, cytosine, thiamine is T, and uracil. So that is there in the RNA. I'm not going into the much details, just to give you an overview. Like these are the molecules that actually are distinguished within the DNA. So those distinguishing features then give us these 4 alphabets with which this DNA is made up of. So we can call them as alphabets, and we can take the DNA as a book of life, right? Our whole genome as a book of life, and this book of life has been coming from these alphabets, right? A, G, and C and T. This is the computational representation of writing some alphabets, like in our binary language. In computers, we have 2 alphabets, 0 and 1, binary digits, bits. But here we have quadruple. They are made up of these 4 things, one of these 4 things. So A, C, G, and T. And then we make words from it, right? So same way, like nature made words from it. So various sequences of the coming from these alphabets, they make your patterns, like promoter sequences, and then they make these patterns, they get together, and they make your further stanzas, like stanzas in a book. So they make further complicated structures, like from the genes you make the chromosomes and other entities. So DNA is nothing but a string of size n derived from these alphabets, right? So that's our computational definition. Watson and Crick gave us the model. Most of you are familiar with this. So here we have the complete structures of those units that make up the DNA. We call them as nucleotides. So they are made up of phosphate, sugar, and within inside of this molecule, we see those nitrogenous bases as we discussed them before, G and C's and T's and A's. So they are here in the middle. On the backbone of the DNA, we see that phosphate, sugar, and that thing runs. This thread runs like this. This goes from here to here, and this way giving it dimensionality as well. So this is the end that is exposed. This we call it as 5' end, because at this sugar molecule that is there in the center, we call it as ribose sugar. So at its position number 5, we have this phosphate group, which is attached. So in this way, this gives this DNA it gives DNA this direction. The other strand runs anti-parallel. So here phosphate, sugar, and it goes, it is pointed upwards, and 5' end here, and at this end we have this 3 position that is exposed. So and this is the DNA's model. We see this red color string or thread, and we see this green color thread. They are wrapped around each other in a double helical structure. So within that, we see that G's on one strand, they attach to C's on from the opposite strand. So G goes with C, T goes with A, C goes with G, Again, G goes with C and so on. So if you have one side of the strand, you can predict or you can make the other side of the strand. So this property was utilized in DNA sequencing, and this DNA sequencing in modern day has been resolved into this, what we call it as next generation sequencing. So we have the overall genome, or the DNA is a larger molecule, a big molecule. We break it into small pieces. We break it into small pieces, and we do that with the help of sound waves. We come up with the fragments of small size like this, large many fragments, right? And then we sequence these fragments with the help of modern day advanced machines. We call them as next generation sequencers, or NGS machines. In this way, when we do that, when we break it into very, very large fragments, we get a huge amount of data comprised of these short fragments. Somebody looked at it like a fire of a shotgun, so he called it as a shotgun method. So you take all those big chunks, big fragments, and break it into the small fragments. So this method is called as shotgun method, right? And finally, you take all these fragments into those state-of-the-art machines. Like one of them is Illumina. These are 4 different technologies. Sorry, giving a very, very brief overview. I'm not going into the bulk detail, but these machines, they have these instruments that do this sequencing for you. So Illumina has the flow cells. So within the flow cells, these lanes or these channels are where you upload your material and it sequences them. We have a lot of primers in them. Within those lanes, we have sections where we have primers installed. So when these DNA molecules, they go inside, they attach to those primers, and then they are sequenced there. The another technology is solid ABIs. Solid technology, this was Illumina. That uses similar thing, which we call it as flow chips. So you see that how we are moving towards advanced technologies. And 454 has a small wells containing a structure called as PTP, pico titer plates. So these plates are where we do this sequencing. And ion torrent, or ion semiconductor chip, and that was introduced in this ion torrent technology. So we do the sequencing in those ion semiconductor chips. So your DNA, as the previous presenter also pointed out, like now it is shifted to those DNA chips, right? And here it is sequenced, and as a result, you get these sequencers are now, they look like this overall. And this is the GSFLX454 Roche company. HiSeq 2000 looks like this, a big one, like a window sized. ABI came up with this solid technology, and this is 5500 XL, the biggest machine. I think now it's retiring. That was there a few years back, and now it's retiring, and ABI is going a little bit back in this market. Illumina is obviously the winning market. This is another ion torrent, ion PGM machine, and 454 GS Junior looks like this. This is a nice fancy bench machine, or a desk machine called as MySeq. So that is installed in various organizations of the country. Okay, here are the pace is going. So now we are moving towards, which we call it as the next to next generation sequencing, or third generation sequencing. So these machines are going like this: a USB size mini ion that is based upon this third generation sequencing technology. The technology is Oxford Nanopore. We do have a structure where we have the membranous structures, and the DNA goes inside, and there is the adapter molecule that is attached and it sequences that DNA for us. And this is of this small USB size. So that is where this space of technologies is moving. And what lies in front of us is various generations of sequencing. The original Sanger sequencing technology was first generation, and the second generation is where we see 454, Illumina, Solid, and ion torrent. And the third generation of sequencers is also coming in with the Oxford Nanopore taking the lead. Okay, so we should better call it as high throughput sequencing. Okay, because it generates huge amount of data. It processes huge amount of data and then generates huge amount of data. And then obviously the challenges are to store this data and to process that data. And for that, we need these modern day techniques like AI and ML, etc. This is how this sequencing output looks like, these nucleotides, and you have to find patterns out of it. You have to find interesting features out from this. The computer code looks like this: 001 001, like these binary codes, whereas the DNA code or organic code looks like this quadruples, like these nucleotides coming from one of these four bases, right? And applications of this technology is, with the help of this, we can sequence the DNA or we can sequence the genome. In case we are doing this, we are in genomics, right? So you can take the full regions, whole genome, and then this is whole genome sequencing, or you can get some targeted regions like targeted sequencing. So there is whole exome if you are taking only the exons or coding regions. So anyways, this whole thing is in genomics. If you are taking the proteins or any other modifications which are attached onto the DNA, we are in the field of epigenomics. This DNA expresses itself with the help of RNA, right? And this RNA is then translated into the protein. So DNA, RNA, and then proteins. So if you are capturing the RNA, you are here. This field is transcriptomics. Transcriptomics, our modern name is RNA-seq. So that is the field where most of this AI and ML techniques are being applied, because RNA sequencing data is huge, is produced a lot. This RNA is controlled by another RNA, microRNA. Sometimes this microRNA comes and interferes with the messenger RNA, and it modifies or changes the gene expression. So this entity is also very important, this microRNA, right? So if you are studying microRNA, you are in Mirnomics. So basically omics everywhere. So we are in omics era. All right. So yeah, so in this, here is the challenge. So we want to read the book of life. Suppose these lines are there coming from Charles Dickens' A Tale of Two Cities, and it was the message that was given to you like this. So he was actually comparing two cities in the World War. On one side, I believe it's London, and other side it's France. So where there is destruction. So he's comparing these two cities, and this was is saying on one side it was the best of time, on other side it was the worst of times. It was the age of wisdom. It was the age of foolishness. It was the epoch of belief. It was the epoch of incredulity. So that is the message which is given to you, right? And we do have spaces. We do have new lines, and we do have quotes and everything. But in real life, when you look into the DNA, everything is like this. Everything is like this. So there are no spaces. There are no new line characters. So this is the message that goes like this. Okay. And when you make its fragments for sequencing, we call them as those fragments as sequence fragments as reads. So when you get those reads, it looks like this, right? And now the challenge is to put it back. This thing is called as assembly. Like you get the fragments, you are having those sequence fragments, you want to put them back. This is a huge challenge. So that is called as genome assembly. So in order to do that, obviously you need some algorithms, some softwares that should do this task very well. And obviously modern day techniques, they are involving AI and ML in it. Mostly we perform these tasks. If we get those sequences, we need to put them back. This thing is called as assembling of the DNA fragments, or assembly, right? So you get those fragments, we need to put them back and make the original genome. So that is one task which we do most of the time. Sometimes we are comparing sequences with one another. You have one sequence coming from one organism, or related organisms are coming from one DNA or one even protein sequence. Proteins are also made up of amino acids. The alphabet size over there is 20. I'm mainly focusing on genomics and transcriptomics in this talk. That's why my examples are mostly the DNA or RNA. So we compare the sequences with one another. This is called as sequence comparisons. So where do we these sequences, they look like similar. There is a match. If they are not identical, it's a mismatch. Sometimes we need to put the gaps in between them. So the another task which we perform here is the comparing sequences. This is here same example, a similar example coming from the protein side. So we are comparing the protein sequences with one another. While we compare these sequences, we sometimes try to put them in the shape of phylogenetic trees. So that's what we do. Mostly these tasks are performed in our life in bioinformatics. That's where we are today, right? Sometimes we are interested in finding interesting patterns in them, like we want to see where this pattern lies, right? So any example for this is, for example, this promoter finding. So if you want to find a gene, you need to find a promoter, and you know that within the promoter, like Tata box and other interesting features or patterns are there. So you try to capture those patterns. In this way, you are doing this pattern finding, right? So gene prediction is a pattern finding job. So here we see those features, patterns, data box. This is ATG is a start codon, TGA is a stop codon. So you found those interesting patterns. Here you are predicting a gene or predicting a promoter as well. So in this case, this is a pattern finding job. So we do gene prediction, we do sequence comparison, we do assemblies, all these things. Now these are our important tasks which we are performing. Sometimes we are determining the structures of proteins, which are the 3D structures, right? So we get it from the sequences. We get the primary sequences and then the secondary ones, and then these are the tertiary structures which we get it from them, that are complicated. So all these tasks, they need AI and ML. And I see that a lot of the projects coming in this domain as well, this protein structure prediction, where people are applying that. Mostly the image processing techniques are there. Okay, sometimes we need to understand and study the metabolic pathways, and that is another task which is there. All right. So challenge is to develop database that represent graphs that can be queried for the existence of nodes. So these various points which are connected from with one another are in the shape of the nodes, and the nodes are put together in the shape of the network. Right. These dots which are connected are nodes. Network is there, made up of nodes which are connected from with one another. So over here we apply various graph solving techniques. Solutions to all these problems is obviously computational. So you need to have computers, you need to learn about these techniques and apply them, or hire somebody who is good with that. So that's where things are heading, because we are having huge amount of data that has been put into these databases. Okay, according to the central dogma of life, the DNA replicates on itself. So this process is called as replication. DNA is made up of A, C, G, and T, these 4 alphabets. DNA, before it is converted into its functional thing, or the functional units, which are the proteins, the actual structural and functional entities, there is an intermediate entity here. So since the cost was low to do this gene expression studies at this level, RNA level, so a huge field called as gene expression studies or RNA-seq analysis came into existence, right? Proteins are still, the protein sequencers are, I believe they are expensive and techniques are complicated. And obviously from one gene, you can get large many proteins, so hard to capture them, all of them. So most of the gene studies, gene expression studies, they are here at this level, right? In the proteins, you have these alphabets, right? 20 alphabets. So they are the amino acids. So protein is derived from these alphabets. A string of size n derived from these 20 alphabets. So DNA, RNA, and protein. DNA, RNA, and protein. Gene expression studies are very interesting. So you capture the RNA, and which RNA we capture, that is involved in protein synthesis, the messenger RNA, right? So you take this messenger RNA, you separate it from rest of the RNAs with the help of poly A tails at the end, and then you derive the oligo T's, the oligos, the run of T's actually, and then these T's, they capture those poly A's. These poly T's are attached to poly A's, and then the RNA is captured. Rest of the RNAs are thrown away. You convert this RNA to DNA, right? This is called as cDNA. The process is where you make this cDNA. This is called as reverse transcription. So transcription was DNA to RNA. Now from the RNA we are making this DNA. So cDNA is made, and then obviously you break it into the fragments, you make the libraries, and you do the sequences. Rest of the things are the things. Remember, the sequencers are DNA based, so you have to have RNA converted into the cDNA, right? Uh, in the gene expression studies, we take the RNA and we get it sequenced and we get those fragments. So when we get those fragments back, we put them or align them back onto the original genes, right? These are the fragments, the sequence fragments of the RNA, and we align them onto various genes. And then finally, we counted that how many RNA molecules are attached to this gene A, how many is that. In fact, these are the fragments, right? And how many fragments are attached to this C. So this way we can count them, and when we count them, we can get a numeric matrix like this in Microsoft Excel or text document, and then you process these numbers. So for example, here in this example, we have the RNA coming from these 5 genes, and these genes are on the rows, and their first column is their gene ID, and then the next first 3 columns are where we have these are the normal ones. We can have one treatment here, treatment 1, and this one is, for example, treatment 2, or we can say it is the normal ones are controls, and these are C means cancerous, and we do it in triplicates, in 3 replicates, right? So we have the numbers here, 436, 536, 776, and then 4, 10, and 3. So obviously it shows that on this side we have the higher expression, right? On this side we have a flat curve, so these 3 have lower expression. So same gene across multiple treatments, across multiple samples, as Dr. Saurabh also pointed out. So this is the RNA which we captured with the help of those NGS sequencers. Then we aligned them with the help of some softwares, and then we counted these numbers, right? And then we can throw away some statistical test over here like T or any other test, and we can get a p-value. That p-value tells us like whether these genes are significant or non-significant. If the p-value is less than 0.05, like here it is 0.04, this gene is significantly differentially expressed. When you say differentially expressed, there is the difference in expression, a significant difference in expression. So expression was higher in the normal cells, and in the cancerous cells it went down. So this gene is basically where the expression is gone down, and the genes are down regulated, right? With the expression is going up, the gene is up regulated, up and down, right? So these are the differentially expressed genes which we actually get from this. Okay, so that was the background which was needed up till now for further studies, for further, these lectures. So now we switch gears towards the artificial intelligence and machine learning, a brief overview of that, and in a couple of examples, I would show you how we applied this in our work. Okay, so machine learning is, before we go into this machine learning, originally the field started as artificial intelligence. Some people say machine learning is part of it, but now the modern day definitions are there where ML is the big entity and AI is part of it. But anyways, I'm not going into that discussion. The branch of computer science that has been born at a conference held at Dartmouth, USA in 1956. The scientists attending that conference represented several different disciplines: mathematics, neurology, psychology, electrical engineering, etc. And they were trying to use the recently developed computers to simulate various aspects of human intelligence. So they wanted to have the intelligent machines. Initially, the machines were more like interactive, like the talking machines, or the something like vending machines or ATMs and these kind of things. But gradually, they started putting the intelligence in them. Artificial intelligence may be defined as the branch of computer science that is concerned with the automation of intelligent behavior, right? So various definitions which I get it. Intelligence demonstrated by machines. Someone defines intelligence. It is the intelligence demonstrated by machines. It refers to the simulation of human intelligence in machines that are programmed to think like humans and mimic their actions. AI is being tested and used in the healthcare industry for doing this dosing drugs and different treatments in patients, and for surgical procedures in the operating rooms. They also developed the computers that play chess, and one thing happened was that was the biggest achievement. Like this chess computer defeated the world top player, and that was, everybody got attracted towards AI and machine learning. So that was the big achievement. And self-driven cars. You might be familiar with this TV serial that was back there when I was a baby at that time, by the way, but still I remember that on PTV it was telecasted, Night Riders. So there was a car that was talking to this gentleman. His name was Michael in the TV serial, actually David Hasselhoff, right? And now we are here. That was at that time it was an imagination, or it was you know it was where they were aiming at. But now we see there are the cars that are coming with this intelligence. All right, and detect and flag activity in banking and finance, such as unusual debit and usage and large accounts deposits, etc. So it's there. To streamline and make trading easier, this is done by making supply, demand, and pricing of securities easier to estimate, right? Okay, so various types of AI. Weak AI is a system designed to carry out one particular job. Weak AI systems include video games, chess, and personal assistants such as Amazon's Alexa and Apple's Siri. You ask the assistant a question and it asks you. Basically, these are the interactive machines, right? So you ask a question and it answers to your questions. You might have played with the softwares that talk to you, so they come under this thing. So you have already embedded some patterns, some answers in it. So in the coding, they put multiple answers in it, and then when the user interacts with that or talks with it, it gives you one from that set actually, and then with the next question. So in this way, it engages the user, and you keep on enjoying the discussion. But in the strong AI systems, they are human-like. They are more complicated, and they have some thinking ability in them as well. All right. Then what is machine learning? So machine learning is the study of computer algorithms that improve automatically through experience and by the use of data. So probably we classify them in this strong AI applications, right? So here you have some learning from your data set. So it's a branch of AI, I would say that, but some people still believe like ML is now the big entity and everything is included in it. So based on the idea that systems can learn from data, identify patterns and make decisions with minimal human interventions. Okay, so you feed the data, system goes and does it by itself and brings you some interesting patterns. Okay, sometimes you feed the system some model to work upon, and you give him some training as well, right? So they are classified with the help of whether the training is there or whether no training is there. I'll come to that. So basically what we are doing over here is you are applying some training data, and it is getting some features from it, some variables from it, and then it is applying some algorithms, and it is getting working for different models which are suited to that. Sometimes we feed him which model to apply. This is the training set, and then we give him the actual data, the test data, right? Similar happens, model applies, and it gives us our predictions. Sometimes we apply multiple models, and then we get about various scores for them and find the best fit model. Okay. Various types of machine learning. There are three main types, though there are many, 14, 15 I have seen in the books while I was preparing this lecture. One is supervised, one is unsupervised, and one is reinforcement. Supervised, unsupervised, and reinforcement. Supervised is where it learns through examples. So labels are given, and then we get the desired outputs, exactly what we want. So initially we tell them give them some learning. Unsupervised, it learns from the output. Labels are not given, and this does not give us our desired output. I will clear in a moment. Useful for learning structures in the data. So you just feed him the data, and then it finds various structures or patterns in it and gives you the output. All right. So in supervised, you give him the training set, and then you give him the test set. In between, you also tell him which model to apply. So there is a supervisor here. Obviously you are doing this, but within that there is some algorithm that is operating. So that algorithm is kind of a supervisor in it. In reinforcement, an agent interacts with the environment and learns from that interaction. So environment gives the feedback that may be positive or negative. All right. Applications in which supervised learning is applications in which the training data comprises examples of input vectors along with their corresponding target vectors. This is known as supervised learning. So this definition comes from pattern recognition and machine learning, this book. And the term supervised learning originates from the view of the target Y being provided by an instructor or teacher who shows the machine learning system what to do. So within that, there is some algorithm and that is operating and it is behaving like a supervisor. Supervised learning is the models are fit on training data that is comprised of inputs and outputs. And then these models are used to make predictions on test sets where only the inputs are provided and the outputs from the model are compared to the withheld target variables and used to estimate the skills. I'm sorry, a little bit boring and too much lingo here. Their main classes and categories. So there are two main types of supervised learning problems: classification and regression. In classification, we involve predicting a class label. We involve predicting a class label or basically a variable that is categorical. So for example, you give some weights of some individuals and then you see whether these weights, with the help of these weights, can we predict the gender of some individual. Obviously, males have more weight than females, so maybe you can take the weight data of various gentlemen and then some ladies and then you put it into the system and then he trains on that, and finally when you give someone's weight he might guess or judge his gender. So that is the classification. Regression, where the supervised learning problem that involves predicting a numerical label, right? So age corresponding to weight. So what should be the weight of somebody at this age and that age and so on. So both the variables which are correlated here, they are numerical, right? So classification and regression problems may have one or more input variable, and input variables may be any data type such as numerical or categorical. But the output is that is the decisive thing. So the thing which is predicting is called as predictor, right? Predictor. The thing that is predicted is I call it object here. So that is whether numerical or whether categorical. If it is categorical, obviously we are going towards classification. If both are numerical, like in this example, we have on one side gender and on other side we had the age or we had the weight. So weight was the predictor. So we were predicting or we are getting actually one of the two classes, whether male or female. So in that case, it becomes a classification problem, right? So you are predicting a class label. This thing is class label. There are two classes in it, right? Male and female. Or sometimes you are predicting the nationality. So again, it's a classification problem. In regression, both the things are, especially the things which is to be predicted, is a numerical thing. You are predicting a numerical label, numbers. All right. Again, classification. An example of classification problem would be the handwriting recognition system. So you give various handwriting text and then it recognizes. Should I take the questions in between? I think maybe we can go in the end. So handwriting digits are given and then you can recognize whether it's a number between 0 to 9. So there are various classes and then it puts them and assign them. So I, for example, write 0 and then there is a system that takes processes this and brings me a digit 0 like this to the computers, right? So I just write something which is something like this, and he says, and he processes it and then classifies it that I have written the digit 3. In regression problem, a house pricing system where the inputs are variables that describe a neighborhood and the output is the prices. So you give him even the input maybe, or in the predictor you might have categorical or you might have continuous, but your output is always a continuous thing. So he's giving you the prices, the numbers. All right. So the ML machine learning has the algorithms. So algorithms are referred to as supervised because they learn by making predictions and given examples of input data. And the models are supervised and corrected via algorithms, right? So algorithms, we decide them, which to pick, and we feed these to the system. And then the algorithms, they take control of the everything inside. So yeah, the predictions themselves. So in this way, it is all supervised. Some algorithms may be specifically designed for classification like logistic regression or linear regression, and some may be used for both types of problems with minor modifications like ANNs. So Dr. Ahmed will cover ANNs. I would briefly describe how linear regression works and then switch gears towards the other methods and a couple of other examples. All right, so linear regressions finds a way to correlate each feature to the output to help predict future values. So at first we try to correlate the objects with one another, the variables with one another. In logistic, we actually classify them, right? The output variable is, as I said, binary, black or white or something like male or female and so on. Decision trees is another type of supervised learning where we have this highly interpretable classification or regression model that splits data features into branches. So you get a tree shape and you can have various decisions at various branches. So you are moving in the shape of a tree. You have the nodes down there, the internodes, and the terminal nodes or leaves. I'll show you in a moment. And SVM is sport vector machine where you uh, take up your data set and then you classify while imposing a plane between them. You draw a line and then you have convert that line into a plane and then you separate the data into different classes. So that is the sport vector machine. All right. So before we go into these complicated models, I would just briefly give you an idea how this linear regression works because it is very simple and easy to understand. And probably if you get it, then rest of the things are built upon this. They are quite complicated here, not to be covered in this section, but you should know how the things work. They work in similar. So variables can be classified into dependent and independent. Whenever we do data collection, we do it in the shape of the variables like age and gender and weight. All these things are variables, right? And sometimes these variables, they start dependency on each other. So one type of variable is classification or independent variable, and the other one is response. So for example, here I can give you the example where you study for some exam. So hours you study for some exam is X, and your exam score depends on that. So Y depends on X. Can we see a relationship between these two things? How many hours you study and their grade is, if you look into the data points, I think we fit in a line here on this in these data points, right? So this line can be squiggle or this line can be a straight line. I'm sorry, something wrong with this. So we can set up a straight line like this. We can set up a straight line like this. We can set up a line like this. The best fit line is that has the smaller distance from these data points. So obviously this straight line going up is the best solution, right? So we can fit a straight line. Best fit is straight line going up like this. We do have other lines as well. So that's our model, right? So in this situation, we can apply a model. This model is linear regression. We set up a line and then we do some calculations here and we calculate some other important things from this. Example is we have some data set where we have some students and these are their scores. So their interest in theater and interest in retailing and maths and language. And we can see how these various interests they correlate with one another. So math versus language, we see something like a straight line. This thing is over here we can plot math here on x-axis and language on y-axis, and most of the cases we are looking for y that depends on x. So y here, x here, dependent variable, and at the x we generally put the independent variable. So language aptitude depends on maths, right? Looks like we can fit up a straight line going up. Math versus theater, we can fit a straight line, but not that good. So probably the model is not that accurate. In the first case, it is good. Here, a straight line going down may be a best fit. Here, again, maybe a straight line going up, but again, score will be low. So we are fitting those lines. We are doing some calculations. Those calculations are what we call them as models accuracy or performance. And finally, we are deciding whether we are fitting a good model or a bad model, right? First, we find what we call it as a Pearson correlation. So that's how it is calculated. I'm not going into the detail of it. Linear regression. So this Pearson correlation are all small letter R tells us how these two things are correlated with one another. Its values, they go from 0 to 1, 1 being the perfect correlation and 0 being the poorest correlation, right? So if the things are correlated with one another, then we move forward further and do apply this prediction or regression. In linear regression, the idea is to use data on an independent variable to predict or explain variation in a dependent variable. And we can calculate various entities like what we call it as intercept, the place where this straight line touches the x-axis, like where the x-axis is 0, the place where it touches y-axis when the x is 0, right? So both the things, they started some relationship, but y was already at some point when this relationship started. So I give the example like when a baby is born, it has some weight. It's not like 0, 0, right? The age is 0, there is some weight that is already there. So that thing in the model is here, it's called as intercept. And then there is another thing is when x changes 1 unit, how much y changes. That thing comes it in this beta 1, which is the slope, the height of this or steepness of this line, right? And we calculate these things with this ugly equations. I'm not going into the detail of it. If you want to look into the background, I can give you these slides. So here this is an example like sales depends on purchases. So your sales are good, then you apply for more raw material and you will do more purchases. So how do those sales, they depends on purchases. We put them, and this how we calculate them, right? And these all calculations are there in this table. So finally we get this equation. So there is some factor already there which is called as this intercept. I'm sorry, I'm short of time, so I need to rush a little bit. And then this is where the slope is. So every 1 unit increase in x, y increases twice, 1.7 times. This is where actually they started, right? So we can predict what will be my sales when I have this x at 15% or 15, right? So 15 pay, we can calculate what will be my y if x is 15. So that is, we calculated as 34 point something. There is another thing that's called as R square. So that tells us how good is our model. R square is how good is our model, and that is the goodness of a model. This is how we get it in Excel. So our R square is 0.95, close to 1 is good. So we fitted a linear regression over here between these two variables. This was our slope, and this is our intercept. It goes other way around. R square is 0.95. So that means this model is 95% good. So as I said, 1 is the best and 0 is the worst. So 0.95 is close to 1. So it is the best fit model. So we fit a best model over here. So in this way, we can get the accuracy or fitness of the model as well. Another example, global warming versus or the world temperature versus the melting of the glaciers. So we see a straight line going down. The model we fit has R square of 0.702, which is a little bit not that good as the previous one. All right, so in the logistic regression, I'm not going into this, but here is where I want to focus upon. So on this side, for example, you have the weights of various individuals or ages or any other features. And here you are predicting whether it's, for example, it belongs to class A or class B, right? So up till this point, for example, here are female or male. So here your dependent variable is actually a binary thing, one of the two things. So in this case, this where you will fit this logistic regression, right? Equations are a little bit ugly and complicated. Decision trees are where you make some decisions like this, like one of the two things. So weather, how is the weather? Sunny, and the time for the bus is, it will take more than 30 minutes. I think I would prefer walk. It's sunny. The bus will take less than 30 minutes, so let me take the bus. It's cloudy and I'm hungry. So yes, I'm hungry. So I think let me go on a walk and hit some restaurant as well. So I would prefer walk. It's cloudy. I'm not hungry. Let me take the bus. Rain, for sure, I will take the bus. So here you are applying some decisions here. You are taking some decisions here. You are in a situation where you need to opt one of the two things. Support vector machines again separates the data into different groups and classes. So you try starting applying different vectors originating from the same point, and then finally you get a plane. This is a line, and these two widths then make it a plane. So you fit up a plane, or a plane that is best separates the data points into various classes. So that is a example of a classification thing, and it is the support vector machine. In unsupervised learning, and let me wrap up within a couple of minutes, uh, you don't have this supervisor, or you don't give any training or testing. You give data everything, and then data clusters the things by itself, right? So within that, we have this clustering and density estimation. So, and then within that, there is one thing which is the k-means, where you give them some, you determine that how many clusters I should make. Sometimes, but most of the time, system does it by itself. So here is one of our data set where we have those RNA sequencing data coming from various varieties, three varieties of soybean, and these are in three replicates. The varieties are NARC and Raval and Savat, and these are their densities. So we see most of them, they are consistent with one another, except this Raval is slightly going off. But this density, kernel density function does not give us a clear separation. So we apply the other methods like principal component analysis. So you give the data and you ask the system to run PCA, and system separates the thing itself. You don't need to give any training or testing. So it separated our data set like this. So here we can see clearly like our various replicates of various varieties like NARC are together, Raval one are together, but in Savat, two of them are together. One of them is really off. So we might think of, messing up here while we were doing the experiments, or while we were extracting the RNA from this variety. So this thing is again unsupervised learning. It's one of the machine learning thing. Most of you, I think, who are doing this analysis, they are applying PCAs and all those things, but probably you are not aware that you are actually doing this machine learning, this unsupervised machine learning. Reinforcement, I'm not going to focus much upon that. So here you have some feedback, here you have some reward system. And one of our work where we applied this is we had cancerous data, we get it from TCGA. We get like over 9,000 tumor samples and 741 normal samples. And finally, we get differentially expressed genes across all 17 among those types. Total types we have 24 cancer types, but for some we don't have the normal control, so we left them. So finally we were up with 17 of them and 180 genes that were differentially expressed all against all of them. This is a heat map which Saurabh was also showing. On this side you have the samples, the type of cancers, and on this side you have the individual genes, and you can give it to any classification system. So here we already did that. So all the genes that are upregulated, showing the red colors, high fold expression in the cancers as compared to the normal ones, they are up in the upper part, and the green ones are on the bottom part. We can apply this hierarchical clustering as well. That is also one of this machine learning thing, and in this way we can separate the cancers that are having the similar gene expression patterns. So we can make their trees like this. We applied k-means clustering, that, and then we partitioned the genes into various clusters or groups. So mostly the genes like tumor suppressors, they went into one group, and oncogenes among cell cycling and signaling, and all the other pathways. So we have seen this thing applies there. So similar genes have the similar expression patterns. Last but not the least, we applied predicting gene expression using machine learning models. We thought maybe this DGE testing, normally it is done with the help of applying statistical tests. So we did that before. and then we knew what genes are differentially expressed. That was our reference. We call them as the actual ones which are differentially expressed, and we thought, let's try with this various machine algorithms, machine learning algorithms, how would they do that? And we checked their performances. This matrix is called as the confusion matrix. So decision tree and logistic regression and k nearest neighbors and SVV, see, we all applied them over here. And this confusion matrix tells us like how good we are in predicting. So for example, here in this section, we have the positives that are supposed to be positives. So these are our true positives. And these are our, this is true and false, and this is true and false here. So true negatives and false negatives. So some of them, they were declared not differentially expressed. So these are our false negatives. When we applied various methods, we applied like these true positives were same for all of them, but and similar pattern was observed in the false negative. But these methods, they work good as well, but still we are improving on their accuracy. All right, the host is here, so that's a warning to wrap up. Same way we did this training on these, and we observed this random forest classifier worked well for most of them. Right, in testing accuracy, again, random classifier training in our results for these three cancers, they were doing well. Thank you so much all, and this is all for classification accuracy. And wrap up, that's what this machine learning is all about. I hope it will pave the way for understanding the things which are coming in the next very interesting lectures. Yes, over to our host.
Thank you, Doctor.
I can go and answer to your question in the chat box as well. Right.
But we'll open the floor for only three questions before we go to the next presentation. So we have a raised hand from Khurram Shahzad. Mr. Khurram, you can ask the question.
Khurram, I think he had an issue with the hearing. He says unable to hear you. Please check your mic. Ji Khurram, in addition to that, if you have any question, please. New message. Noman, he says thank you.
Do we have any participant who wants to ask a question from Dr. Wasim? I guess then we should move on with the next presentation.
I'm taking them on the chat box as well. Yeah, go ahead, please.
Sure. So we'll move on to the next presentation by Dr. Aziz Khan. He's from Stanford Cancer Institute, and his topic is application of machine learning in regulatory genomics and epigenomics. So over to you, Dr. Aziz.
Thank you, Harsh. Let me share my screen. So can you see my screen?
Yes, sir, it's visible.
Okay, so before I will move to the main topic, I think I need maybe 10 to 15 minutes to go over one of the very important topic for the current situation that we are all going through, and also, of course, for the machine learning and AI, which is open science and open data. And again, I will thanks to Dr. Wasim for inviting and also giving a very useful primer on AI, ML, and life sciences. So I don't need to bother going to more details in my next talk. So again, open science such as open and fair data are central for development of machine learning and AI frameworks. We have already witnessed the importance of open science from the current pandemic that we all going through, from sharing the virus genome data and sharing all the science publicly available right away. So it is important to do science, of course, but it's very important how we do it and share it. Science is incremental and becoming more collaborative, and that requires of us to make our findings public in order to lay the foundation for the next level of discovery. But for many of us, especially the young researchers, that we think that publishing is a race, rather building it together. We compete against one another. However, whatever the way you think or look at the problem or at the science, speed is beneficial for both science and scientists. The faster new discoveries are made available, the faster they can contribute to further findings. But the traditional way of publishing science is very slow. It takes months to years to get a paper out. You submit to a journal 1, it gets rejected, journal 2, and then so on until you will find the final journal which is agree to publish your research. It takes years to get it to the public. But preprints are like manuscripts shared by scientists. It is the best way to address the challenge by posting preprints. Preprints been known in other disciplines, especially in physics, for decades now. So the preprints are fast, free, and open. So instead of going through all this, you just post a preprint to a public server, which can be online in less than 24 hours. And you can get feedback from the community while your paper is in review. And you can make your science public very fast. And of course, there are some concerns over the preprints. The first one is being scooped, that you put a preprint and someone stole your idea and get it published before you. But I think that's not the case, because by posting a preprint, you have a timestamp on your claim that you were the first. And the other thing, maybe it's against the journal policy, but that's also not the case, because hundreds of journals now accept preprints. And there are some journals even you can submit directly from a preprint server to the journal, which is called B2J. You can find a list of these journals here. And the preprints are increasing in the life science, especially. Like in 2019, 56,000 preprints been posted, and the growth over 4 years is 1,000%. And there are several preprint servers out there, from bioRxiv to medRxiv and so on. And again, the preprints played a key role in the current pandemic. More than 3,000 preprints were posted on bioRxiv, and also more than 11,000 preprints were posted on medRxiv. And preprints comes with several benefits. It's open. Your science is open for everyone to access. It gets visibility. You get more citations and increase the impact of your science. And of course, it advances your career. How it advances, instead of saying my paper A is under revision in any of the top journals or any journals, but no one will care. If you have a preprint, just provide a DOI. It can be a useful tool to get hired, to get a collaboration, or even a journal editor can approach you and say, oh, hi, would you like to publish your preprint in our journal? And of course, many funders are now requiring and also encouraging researchers to post preprints. Of course, you get more community feedback. You post a preprint, you get comment, and you updated your work, and you post another version of the preprint, and then you will publish it. Journals are already using as a marketplace. Some journals even have preprint editors. For example, PLOS Genetics, and you could see on Twitter, people are approaching if you post a preprint, and editors are saying, would you like to publish in our journals? And for young researchers, especially in the global south, where I see there is quite this disconnect in terms of open science and preprints. I think preprint journal clubs are very good way to train your trainees to review these preprints and post on the preprint journals on preprint servers such as the prereview.org, where you can review a preprint and put it up as a publicly or send it to the authors. So ASF Bio is an organization. It's a nonprofit driven by biologists, which works to enhance the awareness of preprints in the life sciences. You can find more resources on their website, and of course, there's a huge community involved to raising awareness about these preprints. And I see there is no one currently from Pakistan, especially very few from the global south. I would really encourage you to be part of. If you are interested, please reach out to me. And again, for young researchers here, I would like to share this great resource we put together with the help of eLife. We call it ECR Central, or Early Career Researchers Central, which provides funding opportunities and also travel grants. Well, travel grants once we are done with the pandemic, of course, and also a community for discussion and so on. So please do post preprints. You can cite preprints, of course, and you can review preprints, and of course, all of us, we read preprints. So thank you. This was the talk about preprint. I'm happy to take any questions if you have about the preprint part. No. So let me go to the main talk. Okay. So back to the genomics. So again, thank you, Dr. Wasim, for inviting and also giving a very thorough and detailed primer on machine learning and also part of the life sciences part. So I will focus on mostly on regulatory genomics in the non-coding part of the DNA, and I will share some of the current state-of-the-art methods being developed for the regulatory genomics, and share also part of my work and some of the resources that we developed could be used as a to train your machine learning algorithms as a data. So we, the Homo sapiens, are made up of trillions of cells and of hundreds of types, of course. Each cell in our body carries the same DNA and around 20 to 25,000 protein coding genes. In any particular cell type, there are nearly 14,000 genes expressed, and remarkably, only 3% of these genes are true cell type specific, and majority have housekeeping functions. How these small fraction of cell type specific genes turn on and off in a particular cell, that is driven by a mechanism called cis-regulatory regions or cis-regulatory elements, which are part of the non-coding part of the genome, which is most of the genome is non-coding, which is 98%. These cis-regulatory elements are enhancers, promoters, insulators, and silencers. So today I will focus more on the enhancer part. So enhancers are cis-regulatory elements present in the non-coding DNA, which has the binding sites for transcription factors, which not only enhance the transcription of the gene, as the name suggests, but also play a very key role in cell type specific gene expression. It's been more than 30 years since we know the first enhancer. So you could see from this cartoon here, this is the DNA looping around, which this looping mechanism is mediated by the cohesion complex. So enhancer B coming close to the gene X to enhance the expression of the gene X, while enhancer B is inactive. So this enhancer is A specific to this cell type, while enhancer A is not. So I love this picture. It's somewhere up in Gilgit-Baltistan, where I come from, which explains quite well gene regulation. So you could see this road as a DNA looping around, and the green part is the active region, which is compartment A in the chromatin structure, we call it. And the other part, across the river, you could say compartment B, which is inactive. And the river, you call it these topological associated domains. These are the boundaries which create these compartments. And of course, these little two red houses, they are, I would call it the CTCF anchor points, which makes this loop. And the rest is all transcriptional machinery and transcription factors, which are coming into the play the role to express the expression of the gene. So again, back to the enhancers. So enhancers can be active. For example, right now here this enhancer B is active, while enhancer A is poised. So there are histone marks and also other key regulators known to bind or to mark these enhancer regions, like H3K27 acetylation is known mark for active enhancers and also monomethylation, and H3K27 trimethylation is a mark for poised enhancers. So over past couple of 2 decades, or there have been tremendous development in terms of methodology and also technology to dissect these regulatory elements in the human and also in mouse genome. So the ENCODE consortium played a key role to creating several types of assays on different levels of the biochemical features of the DNA. So from chromatin structure, like the 3D structure from Hi-C to ChIA-PET, and then the open chromatin, the ataxic, which is currently is state of the art, and also the DNA-seq and the ChIP-seq to find the binding site for certain proteins in the DNA, and the RNA-seq, and so on. And also using this data, there have been a lot of computational methods, machine learning based, to predict these regulatory elements throughout the genome. So here you could see these different regulatory elements. So using these techniques, several international consortiums like the Roadmap Epigenome Project, the ENCODE, and the Phantom generated lots of data in hundreds of cell types to map these regulatory elements. So there are 1 million enhancers in the human genome, around 10,000 in a single cell. So but the identification of these cell type specific and disease specific enhancers is challenging because there are not really clear known features. So where, of course, the deep learning and machine learning comes in to understand through the black box, which we really don't know what's going on in these deep learning models, but it does an amazing job. So the ENCODE team put together all these regulatory elements from enhancers to promoters, using all these public data being generated by ENCODE and other consortiums. They call it a screen, which is very useful website and tool to go and look for these regulatory elements and use for your analysis, and also develop models on top of that. And another effort at Stanford recently been by William Greenleaf and Harvard Chung's lab, who they developed the ataxic assay. They profile cancer, 23 cancer types from the TCGA atlas for 410 tumor samples. So, and then they've found 562,000 regulatory elements in these cancer. You could see that they are cancer-specific regulatory elements. So using, of course, the data from ENCODE, Roadmap Epigenome, and several other consortiums, there have been a lot of machine learning methods being developed, especially the deep learning for genomics and regulatory genomics especially. So the DeepBind uses, again, DeepBind is a deep learning framework which uses high throughput genomic data to predict the sequence specific specificities of the DNA, and also the RNA binding protein. I will not go in detail in these models, how they did it, but please feel free to. I will share the slides if at the end of the talk if you're interested. So another one is the DeepC, which is again a deep learning based algorithm predicting the chromatin effects on sequence alteration. You could see, and you provide a sequence with alteration, and it uses again the data from ENCODE and Roadmap Epigenome to predict the effect of this alteration on the chromatin in terms of DNS hypersensitive sites, TF binding, or histone marks. Another one, which is called Basset, again, it's deep learning, which uses convolutional neural network and genomic data to annotate and interpret non-coding genome. This is recent one from Anshul Kundaji's lab at Stanford to decipher the regulatory grammar of these cis-regulatory elements using just sequence alone. So you could see that from this key regulator, the OCT4 in ESOL, this is the observed profile, and this is the predicted one, and this one is for SOX2, and this one is for Nanong, and this one is the scale of 4. It's quite surprising and amazing that you don't need an assay even to profile these. You don't need a chipstick to profile your protein of interest. You just use the sequence. So this is another one from the same people from the same lab where they did the, as Dr. Wasim mentioned about the chess one, the AlphaGo. So they recently, it's been breakthrough in the protein structure prediction. So they call it AlphaFold, which predicts accurately predicts the 3D models of protein structure, and of course has very potential to accelerate research in every field of biology. So this is a great resource I would like to share. It's called KaiPai. It's a repository of ready machine learning models for genomics. You could see many of these, some of these I mentioned, and also several others. They are there, they're already trained, and you can just use them. So this is until now is the kind of current development in terms of ML in regulatory genomics and epigenomics. So I will go back to the enhancer. So as I said, enhancers are cis-regulatory elements. They enhance the expression of the gene. They are in the non-coding part of the genome. So if you look at the SNPs from the GWAS, remarkably, majority of these SNPs from GWAS are non-coding. And other surprising one is that most of these SNPs, they are either on enhancer regions or nearby the enhancer region. So this plot, you could see the distance from the enhancer. And a small group of these enhancers, they cluster together, and they form these more, the cell type specific enhancers. They call it, well, Rick Young's lab, they call it super enhancer, which the name quite controversial in the community, of course. But there've been a lot of studies and following the potential of these super enhancers in biology, and also even there are some pharma companies started using these as a transcriptional drugs to treat some of the rare diseases. So these super enhancers they are cell type specific, and these super enhancers again control cell identity and disease. The other great feature of these super enhancer regions are they are very sensitive to perturbation. So here you could see in multiple melanoma cell, you try to disrupt this super enhancer region using a transcriptional drug, which is JQ1. And you could see after the disruption, the expression of the oncogene MYC reduce significantly. So during the same time, there was another concept that from Francis Collins' lab at NIH, they come up with this the term they call it stretch enhancers, which are again large enhancer. These are defined based on the size only, but the super enhancers, they are defined based on the signal of certain biochemicals like MED1 or STK27 acetylation. Again, these are also cell type specific, but there'd been quite a confusion among the community. Some people were calling it the SEs like super or stretch enhancers, but they seems quite significantly different because, of course, the way you define it's totally different. It's here just based on the size, and here you have the based on the signal. For example, the MED1 signal. So they use this cutoff where slope is 1. They call it super enhancers. Of course, the definition is not the biological one. It's totally mathematical. So we looked at, again, using public data from ENCODE to see the differences between these super and stretch enhancers in 10 different cell types. You could see that the blue one, they are the super enhancers, and these orange one, they are stretch enhancers. In terms of the expression, they are highly expressed, the super enhancers. The genes associated to these super enhancers are highly expressed. Of course, how you associate a gene to an enhancer or a super enhancer, that's another big challenge in the field, where, of course, the machine learning and deep learning will play an important role. Here, the growth signal, where you could see significantly higher in K562 compared to the stretch enhancers, and also some of the active marks like acetylation, monomethylation, and so on. So monomethylation is the enhancer mark that makes sense, like we have both in super enhancer and stretch enhancer. Yeah. So they was published in epigenetics. So if you are interested, please find more details there. And next, we look at, we were interested, is it possible that we could use publicly available genomic data and epigenomic data to predict these super enhancers using machine learning model, and also identify key features of super enhancers. So we used several state-of-the-art algorithms, of course, and try to predict these super enhancer regions. This is in most embryonic stem cell. You could see that random forest is performing well, which is an ensemble method to predict for prediction. So I will not go through, of course, what random forest is. Dr. Wasim already covered that, and also several other machine learning algorithms. And we validate the model using in other 4 different cell types. This is totally independent data set, not seen by the training data. It performs quite well. And by looking at the feature importance of these super enhancers, we try to rank. So we use one back out approach. We take one feature out and see how the performance loss. So they call it a one back out approach, and to to compute the feature importance. So here you could see that, of course, STK211 oscillation was already known but reported by Rick Young's lab, and also BRD4 as a key feature. But we also found CDK8 and CDK9 as a key feature. And we try to, again, use these features alone to predict these super enhancers. CDK8 and CDK9 have similar predictive power than acetylation and BRD4. And this one in ProBSol, you could see that SMED3 turned to be one of the key regulator. This is a factor for TGF-beta signaling pathways. And again, SMED3 was better predictor than ESRB and KLF4, which was known from before. And we try to predict, again, super enhancer using these features in mouse EL cell and several other cell types. So here you could see that SMED3, so the mediator was MED1 was used at the beginning by Rick Young's lab, and we could see similar performance with SMED3. And once you look at the FOX1 locus in the genome, you have the PO1, which is core regulator of ProBSol, and also SMED3 and MED1. And once you look at the gene regulatory, gene ontology terms for the super enhancers we found using SMED3, they are more ProBSol specific. So in a couple of slides, I will introduce some of the resources we develop, especially for gene regulation. This could be used as a data for ML and AI algorithms. So back to the super enhancer thing. So again, through that method and also Rick Young's method, we predicted and computationally super enhancers in hundreds of cells in human and few in mouse cell, and created a database. We call it DB Super, and it is available in this URL, which is integrated with the UCSC Genome browser, Galaxy, and also the great tool for genomic region annotation. Recently was collaborated with GeneCard. You could see links from GeneCard. If you go to a gene of interest on GeneCard, you could see which super enhancer is associated, and you could get a link back to the DB Super. Another resource some of you must be familiar with, it's very known in the gene regulation community. It's called the Jasper database of transcription factor binding profiles. So Jasper is an open source database of transcription factor binding profiles, or the TF motifs, we call it. So this is an open source. All the profiles are experimentally validated at least one time, and all the data has been manually curated and is widely used and long maintained. It started in 2004 by Albin Sandilin. So I've been part of the 2018. I led the 2018 release of Jasper, and also to the 2020, and we are working on the 2022, which will be soon out. So here you can see the growth of these binding profiles over the time. In 2014, in 2018, since I led the Jasper, we rebranded the entire the resource and then redesigned everything. So it was getting a new look after 14 years. So it was running amazingly for 14 years on the same Perl script. So it has very great, of course, user base throughout the globe. It comes with a powerful REST API to access the data programmatically, and you can download in several different formats. And yeah, and you could find more details in the associated papers. Another resource along the lines is that came out was called a map of direct TF and DNA interactions. Initially was in human, now we extended to 9 species. So these are the transcription factor binding sites. Sorry, we call it unibind. So we have almost 10,000 ChIP-seq data sets, more than 800 unique transcription factors, and so on. So you can visit the website and you can download the data, of course, as flat files, and also you can access it using a REST API we developed. And the preprint recently we posted to bioRxiv. So all these sources I mentioned, they are open, freely available on GitHub. So yeah, but until now what I said was mainly on bulk genomics and epigenomics, but the field is of course shifting now towards single cell genomics and epigenomics. The benefit of it's expensive, of course, to do the sequencing. It's still expensive for smaller labs and groups, but major consortiums such as Human Cell Atlas and several others that they're profiling these biochemical assays at single cell level, which is another era for deep learning to use this data for healthcare and to understand the biology better. So here you could see this cartoon. This is an ataxic profile where you could see this heterogeneous sample. And once you do the bulk, you could see the signal for all these enhancer regions or the active regions, the open regions. But if you do a single cell and then you have this heterogeneous sample and then you separate these samples based on clustering mechanism, and then you could see the profile. These are more sample specific, or the profile of these enhancers. So here you can see that this enhancer is active in this group of cells, but not in this one or not in this one, and similarly for the rest. So we can really capture the heterogeneity in terms of the cell population, and also especially the tumor sample we get, because tumors are very heterogeneous, and if you do multi-region sequencing, you could see that they're really heterogeneous in terms of their genomic and epigenomic profile. So as I mentioned, some of the international efforts that are underway to profile, create these human cell atlases using single cell, the human cell atlas, of course, and also the NCI, the Cancer Moonshot funded, the HTAN consortium, which is we are part of at Stanford, where we are looking at the pre-malignancy and after malignancy, and also pre-treatment and after pre-treatment, and then we create these, excuse me. So we create these profiles or the measurements, you could say, at all the levels, from genome, from genome, proteome, metabolome, metalome, metagenome, and so on. And of course, this data going to be soon public and open to the community. This is a very good resource, of course, for AI and machine learning community to use it for better prediction, and of course to understand the tumor biology. So you could read the marker paper, which was out in Cell recently. So I think that's it. Here is some acknowledgement. Of course, I will thank my mentors and also the JASPer consortium, which we are spread around 6 countries. And yeah, with this, I would like to thank again the organizers for giving me the opportunity. I'm happy to take any question.
Thank you, Dr. Aziz, for your wonderful presentation. So I'll request all the participants, if you have any questions, please raise your hand, or you can drop a message in the chat box.
Dr. Aksa, she has some questions on the chat box, and I would also encourage the students to, ask Dr. Aziz about his journey to Stanford and from Gilgit-Baltistan. Like, a couple of students are very excited to know you are from GB, and mashallah, you went to one of the, obviously the leading institute in the world in Stanford. So kids would like to know about how to follow your footsteps. And secondly, as a teacher, as an academician, would also like to seek some guidance from you as well. What should we incorporate in our curriculum about, tuning into our programs? Because initially when I started bioinformatics in 2005, the things were like sequence alignment and, multiple sequence alignment and phylogenetics. And then in the middle, while I was there for my PhD, it was mostly about NGS and genomics and transcriptomics. And now we are switching gears towards AI and ML. So sometimes we feel like it's like, you're overloaded with many, new things. And yeah, for me, maybe 10 to 15 more years, I would try my level best to cop up with the pace. But for about the youth, we try our level best to, give them the new things as well, whatever we can in our limited capacity. Yeah.
Yeah, I think the field is very moving very fast out. Yeah, and I think it's quite challenging to stay up to date. And now we are more into the cloud era, and yeah, and of course, I spent 6 years in Islamabad and surroundings, so I know the challenges being there, and with limited resources and doing sciences, of course, it's very challenging. So I will be very happy, of course, to connect with your students anytime you want to share my experience, and of course provide feedback and mentor if possible. Yeah.
See, questions, Dr. Aksa?
But you don't want me to hear about my journey, like how I ended
Yeah,
up.
sure.
It's gonna take longer, right? So not today, of course.
I think I met you, I think couple of years back when Zadi was here.
Right.
And I thought you are from, since you did your undergrad from Butims?
No, I did my BSc, and that was like maths and physics at FC College, Lahore.
Oh, okay.
And then I started my MSc in IT at Quaid-e-Azam. And then I moved to, of course, the FAST campus in Islamabad to do an MS. And yeah, working around doing some, I was wanted to be web developer, of course, but ended up going to do a PhD in bioinformatics.
Nice.
And yeah,
and then
moved to Norway for a postdoc. Yeah.
Together. You and Zadi were together in that university, right?
So me and Shujat, we were. So Shujat was my junior at Tsinghua in China, and yeah. So I'm also very into open science, and I really want to create this awareness in the global south about open science, which is very important. So if there are communities in the region on open science which want more international I mean, I would look or want me to showcase them internationally, please feel free to reach. I'm happy to be happy to assist. And also, if there are no open science communities, and I'm happy to start one. Yeah.
Thank you, Dr. Aziz, for your presentation. And yes, we'll hopefully, once the travel restrictions are over, we'll have a physical event as well, and we'll invite all these speakers, including you, to come to Pakistan and give a talk and have interactive sessions with our participants. And for sure, in coming future, we'll have a working group of experts who are working in AI and machine learning in different fields, maybe in life sciences or health sciences. So we can have a group of experts working in the similar field, so we can share knowledge with the researchers as well as younger students as well. So we hope things go better in coming future and travel restrictions are over, so we can have you host you in Islamabad again.
Well, that would be great. Yeah. Thank you.
Thank you. So now we'll move on to our next presenter, Dr. Ahmed Raza Shahid. He's from Comsats University, and he'll be talking about brain tumor segmentation and survival prediction. Dr. Shahid.
Yes, thank you very much. Let me upload the slides, share the slides with you.
Let me take the opportunity, Dr. Aziz. There are some questions in the chat box. If you would like to please answer them.
Sure, I will go over and then address those. Yeah, thank you.
Can you allow me to share it, please, the slides?
So we have given you the right to share.
Because this share screen button is off, so I'm not sure. Okay. Can you please do it again?
Yes, let me check from the IT.
Yes.
Dr. Shahid, please try again.
Let me try again. No, the button is still not working, so I'm not sure if the rights have been given to me to share the
We
slide.
have given you the rights of host.
Okay,
now you can share your screen.
Yeah, yeah, I'm trying to share screen, but the button is not working. So should I try to log in again, maybe?
Or you can either send me the presentation, we can run it from here as well. You can email me the presentation.
Yeah, can you share me the email with me, please?
Yes, yes, please.
In the meanwhile, I think we should open the floor for questions for Dr. Aziz, if any.
Okay, so let me go over the questions. So Dr. Aksakram is asking that preprints are available online, and we fear that our research as a preprint will be copied elsewhere and published. What do you say? So as I said, I mean, preprints are as a timestamp that that you claim you are the first. So you have a better chance to get it published. And also many journals now have, if you posted a preprint, you will not be scooped by someone else, because these are the journal policies that at SF Bio we are trying to work on. And also several journals already implemented these policies that if you put a preprint earlier than someone else, if their research is already published, but then your one won't be scooped, it will be published. So I hope that's address your problem, or the fear of not sharing preprints. So I've been sharing since I started my PhD. Nothing happened, and so many colleagues of mine, they do. But we fear, I think, in the global south for open science, I'm sometimes suspect maybe this has something to do with the colonial era that we went through, that we are not that open. But I think it's there are so many good people out there, and of course you will find some which they do copy and stuff. But again, you won't get scooped. That's there are ways to get it around. Yeah. Any other question? Okay. Well, rest I will go through the chat and answer. And please do reach me with the email, or I'm quite active also on Twitter. So I'm happy to address your questions. So,
all right. Shall I start, please?
Yes, Dr. Shahid.
Yes. I'm Ahmed Raza Shahid. I'm the Principal Investigator of Medical Imaging and Diagnostics Lab, which is a part of National Center for Artificial Intelligence, and it is housed at ComStech University, Islamabad. So today's talk is about basically glioma segmentation, which is basically a brain tumor, a kind of brain tumor. And there are a lot of CS terms here. I'm not sure the full makeup of the audience here, but I shall try to be as concise and brief as possible, so we do not get stuck in a lot of technical jargon here. But still, that's the title of the talk, glioma segmentation, where glioma is the kind of brain tumor which relates to glioma cells within the brain. And we have used an ensemble of 2D, 3D units, and we have tried to do survival prediction using multiple feature fusion. So that's basically the title of the talk. Can you move forward, please? Yeah, so it's based on a brainless paper that we recently published, and it's basically we sent this paper to a Mikai Brass challenge, which is an international challenge where people from all over the world, they submit their techniques, their whatever the techniques they use on the Brass data set, and they submit their results. And it's a kind of challenge. So and we were ranked third place in that challenge. And there are 3 tasks in that. There is the segmentation task, there is the survival prediction task, and there is the uncertainty task. We submitted in the 2 tasks, segmentation task and the survival prediction task, and we were ranked third in the survival prediction task. And the other participants were like people from Harvard Medical School and Mount Sinai Institute in the USA. So they were all highly ranked. And this is our team. We were a team of 5 people who did that. So this paper is basically based on that. This presentation is based on that paper, basically. Can you please move forward, please? So brain tumor segmentation. So first things first. What is a brain tumor? As everybody knows what a tumor is, but still, just to remind everyone that there is normally abnormal growth of cells within the body. It can happen anywhere within the body, and it happens in all lots of different species. Humans are, of course, one of them. And so there's when there's an abnormal growth of cells within the glial cells within the brain. That's what we call glioma, and it's a kind of brain tumor. And some of them are, of course, malignant, and some are benign, which is true for any kind of cancer. And the malignant one is the one which spreads to other parts of the body as well. And non-malignant or benign is the one that is restricted to where it originated. And it doesn't spread to the rest of the body, and it's essentially not that dangerous. So what we do, we are not medical specialists, so we see things from the computer science perspective. And what we have is that we get the MRI images from the radiologist who have marked brain tumor on the MRI images. And then we build AI-based system, artificial intelligence-based systems to try to predict tumor within those images. So we try to predict at the voxel level, and a voxel is basically a 3D pixel. So we try to predict at the voxel level if there is cancer there or not. So that's what we call brain tumor segmentation. So there's a classification task. Classification task would be like if we give a tag on the whole image or on the whole volume, whether this whole volume contains tumor or not. So that would be classification. But if we do it at the pixel level, in the case of 2D images, or at the voxel level in the case of 3D images, that if we classify each and every one of those pixels or voxels, then it becomes a segmentation task. So we do not just tell that whether there is cancer or not, we also tell where exactly it is within the images. Later on, we might also work on grading of tumors. Can you move, please? So these are a few of the statistics which only signify the importance of what we are doing. And brain tumor is not as common as some other cancers, but it's very deadly because it's very difficult to deal with the brain. It's one of the least understood organs within the human body. And there are a lot of people who are suffering from brain tumors. So over 700,000 Americans are living with brain tumor today, which is a big number. More than 84,000 will be diagnosed with primary brain tumor, which originates within the brain. So there's primary brain tumor and there's a secondary brain tumor. Secondary brain tumor is something that originates somewhere else, but it's spreads to the brain, which is equally dangerous. Secondary doesn't mean that it's not that dangerous. It's equally a big killer. But primary tumor is something that originates within that part of the body. So primary brain tumor is something that originates in the brain. And more than 84,000 people will be diagnosed with primary brain tumor in 2021. That's for the US. All these statistics are for the US, but just to give you a glimpse of how things are. And there are more than 120 different types of primary brain and CNS tumors. CNS is central nervous system. So you can also have tumors within the what do you call it? The spine, the spinal cord. And you can have them in the brain, of course, as part of the same system. And there are 120 different types of them. And 1/3 of those tumors are malignant, so they are deadly in that sense. They can spread not just to that part, not just to within the brain, but also to other parts of the body. And out of all these people, 28,000 of them are kids who are suffering from brain tumor right now. And out of all these people, 18,000 are expected to die as a result of malignant brain tumor. And since our work is basically on their survival prediction, on their prognosis, so we need to know the factors that affect survival, and one of them is age. Of course, with age, things change a lot. Geographical location, tumor type, tumor location, and molecular markers. So all these have an effect on survival prediction. Can you next slide, please? So this is the BRATS 2020 challenge pipeline. So we get the input MRI images, which are tagged at the voxel level by the BRATS people. Then we do the brain tumor segmentation task, which in itself is a big task because we have to classify at each pixel or at each voxel level and tell whether there is tumor there or not. And from the segmented tumor, we can get these kind of different segmented tumors, which contains the enhancing tumor, the whole tumor, and the core tumor. Enhancing tumor is the one that is still spreading out. Whole tumor is the one that contains the enhancing tumor, the non-enhancing tumor, which is not spreading, and also some lesions which are known as edema. So that's the whole tumor. Core tumor is anything but the apart from the edema. So enhancing tumor plus non-enhancing tumor combined, they form the core tumor. Then we did feature extraction, which is people who have a background in machine learning, deep learning. You know that is the most essential, it's the most significant part of our job to identify or to extract reasonably good features that can help us classify or segment the images. So we basically figure out different features such as intensity-based features, texture features, and shape-based features, and then we carried out some kind of feature selection. So after getting all these features, we have to identify which ones are better features for which features would help us more in classification or in segmentation, and which features won't be that useful. And at the end, we divide the data set, that is true for all machine learning tasks, basically. We divide the data set into training, validation, and test sets. And that's on the training data set, we basically train the AI-based system and train them on how to identify tumors from non-tumors. On the validation data set, we try to fine-tune the learning. So we try to make learning better. On the test set, we actually test the results. So test data is something that is totally unseen by the system, which it has never seen before, and there are no tags available for them. For the training data and for the validation data, the tags are available. Next slide, please. So what we have done is we have used an ensemble approach. An ensemble approach is where you kind of ask more than one expert about the presence or absence of a disease. For instance, if you are if you're a patient and you go to one doctor and ask him or her about the diagnosis, he or she may tell you something about the presence or absence of disease, but some people would go to another doctor just to get a second opinion, just to make sure that if the first doctor has diagnosed a disease, you just want to make sure that it's really there by asking another expert. So it's a very common practice. There's nothing wrong with that, and we employed that technique a lot in any machine learning or deep learning based work or AI based work. That we do not ask just one expert. We normally train more than one expert on a disease, and whenever a new case is presented, we ask basically sorry about the cough. We ask both the experts about what they think, whether we have the disease or not. So we basically trained two different models. One was a 2D model and one was a 3D model. So these are our experts. So there's an expert who has been trained on 2D images, and there are experts who have been trained on 3D volumes, because in brain MRI, we don't just have one slice. We have 155 slices for each patient in that particular data set. So it's a complete volume, and there are 4 such volumes: T1, T1C, T2, T2A, flare. So these are different. Every patient has 4 different volumes, where one image is of size 240 cross 240, and that's the resolution of one image of one slice. And in each volume, we have 155 such slices. So we have 4 different slices, 4 different volumes for each patient, and we do it from 3 different views. So we do it from axial, sagittal, and the coronial view. So axial is from your jaw up to the top of the head, that is axial. Sagittal would be sideways, and the third one is coronial, that would be from back to forwards. So there are 3 different views, and each patient has 4 different volumes. So basically, since we have the 3D data, so we can apply both 2D models that work on each image separately. and we can also use 3D models that work on the complete 3D volume. Computationally speaking, because we are computer scientists, so we are always concerned about the computational cost of things. So 3D models are very costly. Computationally, they are very expensive because we apply filters on the 3D. Sorry about Dr. Sadiq. I will try to be nearer to my mic, so probably I will be more clear. Please let me know if I'm not clear. So. So we can apply the 3D filters on the 3D volumes, or we can apply 2D filters that apply on individual images. The cost of applying 3D filters or 3D models is very high, so they are computationally very expensive as compared to the 2D models. So we can run a lot of experiments using 2D convolutional neural networks, but we can only perform very few experiments using 3D models. But the results are obviously better with using 3D models, and the reason being that we also have the depth information that we do not use in the case of 2D. So what we said is that 3D gives you better results because you also have the depth information, which you lack in the case of 2D. But 2D is more efficient in the sense that you can do more experimentation with 2D because it takes less time. So we wanted to combine the effects, the advantages of both. We wanted to have depth information that we get from the 3D models, and we wanted to run a lot of experiments that we can do using 2D. So we combined both, and we created an ensemble. So ensemble is, as I said earlier, is like asking two different experts about the same issue, about the same problem. So we have two experts, one trained on 2D images, and another expert who is trained on 3D volumes. And every time we have a new image, we ask both the experts what they think of that, and we take a majority view. In this case, obviously both of them say that this one is tumorous, then we would agree with that, and things like that. We also took a number of different features. We took image-based features, we took radiomic features, and we fused the features and then carry out the overall survival task on them.
Excuse me, Dr. Ahmed, slides are not moving. I see only
yes,
slides.
Because I haven't moved them for quite a while. Okay. So can you please change the slide now? Sure. Yes. So can you see it now, the slide moving?
Yes, please. Yes, please.
Yes, because I was basically talking. It was the same slide, and I was giving you the background on the ensemble model.
Okay, no worries.
So the 3D unit models face challenges like class imbalance, and class imbalance is a very common problem in any such task because tumor, as you know, is a very small part of the whole MRI. So most of the what we see in the MRI is the brain itself, and only a very small, tiny part of that is basically the tumor part. So in that sense, it's highly imbalanced. The thing is that we have to train AI-based models on both the tumor part and the background part, because we have to train the system on how to tell whether something is a brain or not, is a normal brain or not, and we also wanted to be trained on how to tell whether there is tumor or not. So class imbalance is a big issue. There are so many different ways of dealing with that, but we are not going to go into those details right now. And as I said earlier, with 3D units, there's higher computation cost. For class imbalance, loss functions are normally used, and there are other data augmentation techniques. That would be too much detail. What we are more interested in is that since 3D models are very computationally expensive, so even though they give better results, but they are computationally very expensive. So we try to do a lot of experiments using 2D nets which we can do because their computational cost is much less. But we wanted to get comparable results. So there is something called hyperparameter tuning, hypertuning of parameters in the machine learning, deep learning community. There are a number of hyperparameters that define different aspects of the model that we are using. And so we try to play around with these hyperparameters and see which ones perform better. So, but in order to fine tune them, we have to do a lot of experiments. So that's what we did with 2D units. 2D units have less parameters, so they run very quickly. But they do not carry the depth information, so their accuracy is not that high. Next slide, please. So, as I said earlier, we used, for the 3D model, we used the DMF model. And for 2D, we trained them on multi-view. We did multi-view training on 2D models. The networks were trained separately, and then the results were averaged. Next slide, please. So there's a proposed segmentation approach. So we combine the results of both. It just shows that there is a 2D unit on top and there's 3D DMF net at the bottom. We just combine them both. Next slide, please. These are results, and we can see from the results that the median results are high for enhancing tumor. So if you look at the DICE scores, because if you just focus on the DICE scores, that would be easier to see. There is the median score of 0.843 for the enhancing tumor. ET is the enhancing tumor, WT is the whole tumor, and TC is the tumor core. So for enhancing tumor, which is the most difficult to predict, because it has the least number of instances in it. It's the most difficult to predict. But we can see that the median results are better than the mean results. And median results, as you know, that if 50% or more are at a particular stage, then we call it median results.
Hello, Dr. Shahid.
Sorry, there was a big small problem. Yes, so if you look at the DICE scores, the median scores are higher than mean scores, which basically means that on more than 50% of the cases, we perform much better. Enhancing tumor is the most difficult to identify, especially in the low-grade gliomas, as compared to high-grade gliomas, because there are fewer instances of enhancing tumor in the lower grade gliomas as compared to higher grade gliomas. So they are the most difficult to detect. But in our case, our median results are better, so we are even doing better on enhancing tumors. So that's what our results show. Can you go on the next slide, please? And then we computed the radiomic features and dynamic-based features. For ultimately, we fused the two set of features. We combine them together, and we basically try to predict the overall survival. After fusing them, we basically carried out feature selection. Feature selection is the process where you basically choose a few features over the others, which you think carry more information. And by carrying more information, we normally mean that they are more helpful in the classification or segmentation task. So we used recursive features elimination. So we take different subsets of features, and then we run our experiments and see on which subset of features our results are better. And in that way, we ultimately select a set of features which give us the best possible results. Finally, we use the regression approach for survival prediction, because survival prediction task is basically the regression task, because there are infinite different possibilities in that. So we can only use regression techniques over there. Next slide, please. Yes, so here we can see we have got the MRI images, and then we segment the volumes, and you can see the tumors are color coded here, just to show on which particular pixels of voxels we have identified the tumor. And we also take the age of the patients. Can you move forward, please? Yes, and then we take all these radiomic features, which radiologists normally take when they manually try to identify normal from the tumorous region. And here they use normally texture features and different histogram features. And then we combine them with image-based features such as position-based features, surface area, volume ratio. These are intensity-based features which we can directly compute from the images. And then we carried out the feature selection. And after that, yes, please. So these are the results of our survival prediction, the results of our regression. And we can see a very high correlation between the ground truth and our predicted results. Next slide, please. And this is our algorithm. So it's very simple to read. So for each patient, basically, we take the radiomic features, we take the image-based features, we fuse them together, we do carry out selection, feature selection, and then we finally predict the survival days, the number of days a patient would survive once he has or she has been diagnosed with cancer. Next slide, please. So these are different image-based features: volume ratio, surface area. So we try to predict the volume of the tumor, surface area of the tumor. So we try to guess these numbers from the images themselves. Yes, next please. And for radiomic features, we have used Laplacian of Gaussian filter is used. And there are a number of different features that combined form part of radiomic features such as intensity-based features, shape-based features, and texture-based features. There were 14 shape-based features such as tumor volume, surface area, 2D and 3D diameter, etc. 19 first-order features. I'm not going to go into the details. 28 gray level co-occurrence matrix features, which are basically the texture features. And the regions that we extracted features were for enhancing tumor, necrosis, edema, tumor core, and whole tumor. Next slide, please. And these are our results in more detail, which I have shown you earlier. So there is quite a, we can see correlation, high correlation between ground truth and our predicted results. Next, please. And these are our results in more detail. So we can skip them. Yes, next, please. And this is the ranking of our algorithm. You can see in this, there are 3 tasks: segmentation task, survival task, and uncertainty task. Within the survival task, you can see the third ranked algorithm, where it says Mohammed J. Ali et al. And it's ComST's MIDLT, Medical Imaging and Diagnostics Lab at ComST. We submitted our algorithm there for the survival task, and it was ranked third, and the entries from all over the world. And but survival can only be as good as segmentation. So our segmentation must be good enough, but it was not good enough to be ranked among the top 3. But since survival was based on that, so it must be good enough. So there was also a small cash prize by Intel AI, nothing to talk about, but it's something that that's there. So next slide, please. So these are some of the references. You can basically look into them to understand our work more clearly. And that's all from me. If you have any questions, please do ask.
Thank you, Dr. Ahmed Raza. Let me check the chat box. And if we have any questions, we'll just pass on to you. So to the participants, if you have any questions, please do write in the chat box. And we would request all the participants to be interactive with the speakers and ask questions if you have any. So we have a
question about features. Actually, there are a lot of so many different features, and if I go into that detail, that would be that would be another talk on its own. So probably we can leave it for some other time, and if we can arrange some other talk, I can do that.
Probably the variables in statistics, like for example, in our general data sets, we have like gender and age group and profession and something like that. So I think in AI language, you're calling them features, right? Like for example,
They,
it has a shape and it has this much diameter, and yeah, that's what I
yeah.
understood.
Yeah, so these can be some of the features. Normally, gender is not important in brain tumor, but age is. So, because especially in the survival prediction task, age is a very important factor, because the more aged you are, probably survival is more difficult. So, and younger you are, probably you have high chances of survival. So, definitely survival is a lot dependent on age, but it also depends on other factors where the tumor is located. So we try to figure out within the brain MRIs where exactly the tumor is located, the central position of the tumor, its diameter, how big it is, its shape, its texture. All these different factors basically give us information whether it's going to be a deadly tumor or not. And if it's a deadly tumor, obviously the chances of survival are less. If it's not that deadly, or if it's not spreading that fast, then we can say that the survival chances are greater. So all these different features ultimately affect what's the shape of the tumor, how big it is, where it is located within the brain, the age of the patient, and there are so many others. But as Dr. Wasim said, yes, these can be some of the parameters. Yes, we haven't really. Thank you, Jawad Hussain, for your question. We have only did it as a background process. It was not our main task to figure out the exact features which were more useful. We just carried out as a background task. But we took so many different subsets of the features. There were thousands of such subsets that we took. So I can't exactly tell you at this time which features were the most important, were figured out to be the most important. But we can carry out this exercise as a separate experiment, and probably if we get the results, we can share it with you. Thank you. Okay, any other questions?
In case anyone wants to ask any question, we can share the email address of Dr. Ahmed Raza Shahid with the participants, and you can drop an email to Dr. Saab for future correspondence or any cooperation.
Yeah, you're more
So
than
any other
welcome.
questions?
Yes.
So any other questions from the participants? I guess not. So for the session one, all three talks have been done. Now we'll have a short break for lunch and prayers, and we'll resume the next session at 1:30 GMT +5. So I'll request all the participants to be live again at 1:30, or you can stay connected for the next session. Thank you, participants, and we'll